So that'd be just terrible. Okay, there we go. Got the privacy notice. It's happening. Well, welcome to the afternoon physics session for CSURF. Our first speaker will be Cadet First Class Megan Cordone on assessing performance of 2D beta gamma spectrum analysis for radio xenon detection. Megan, it's all yours.
Awesome. Thank you, Major O'Keefe. I'm going to go ahead and start my timer. As he mentioned, my name is Cadet First Class Megan Cordone, and I will be talking about assessing performance of 2D beta gamma spectrum analysis for radio xenon detection. Kind of a mouthful, but I hope you enjoy it. So to start out with the overview of what I'll be talking about, I'll start out with talking about radio xenon detection, kind of what it is and why we do it. I'll then go into some of the existing methods in the field and some of those problems with those methods. Then I'll move into peak fitting, which is the algorithm I'll be talking about here today, the theory and mechanics of how it works, and then how it aims to solve some of the existing problems in radio xenon detection. I'll then move into error analysis and performance comparison, which is kind of my major contribution to the project, and then some other miscellaneous results. So first, to start out with talking about radio xenon detection. So why we do it, why is it scientifically significant? So if you look at those isotopes up there in the title, those are used for nuclear treaty verification. And just kind of speaking broadly, radio xenon, or more broadly, radionuclide detection is used to classify an explosion as nuclear. So if there is a detonation that is supposedly nuclear, you can use a lot of different techniques to look at the size and kind of the different waveforms around it. But using radionuclides or different isotopes from the detonation, those are kind of considered the smoking gun form of detection, because you can have a massive conventional explosion, but if you start to measure radioisotopes from it, then you can kind of check the box that it is in fact nuclear. And that picture you see there is a plume around Southeast Asia of kind of how radionuclides travel, and that's used to detect underground nuclear detonations. So talking about why is xenon an interesting element for this? So first of all, it's a fission product. So if you go to this chart over here on the right, I'll go ahead and turn on my laser pointer. If you go to this chart over here on the right, you can see that on the y-axis, we have fission yield, so kind of the percentage of abundance. And then on the x-axis, we have the mass number, so which element, which isotope we're referring to. So this is for neutron fission of U-235. So you can see this teal and yellow circle up here. That's kind of where xenon falls. So it's a fission product. It's abundant. We know that if there is a nuclear detonation, xenon will be there. It's definitely reliable, which is why we use it. Second, it's a noble gas. So if you kind of remember from your chemistry classes way back, a noble gas is called a noble gas because it doesn't really interact with anything else. So after the detonation, we're not going to see xenon interact with other elements. It's going to stay there. It's going to be stable, and thus it can be used for a detection mechanism. And then last, it has a reasonable half-life. So the isotopes that I mentioned in the previous slide, or that you saw on the top, they kind of range from 9 hours to a couple weeks. And this is beneficial because if the half-life was around a couple seconds, it wouldn't really be useful for collection and then kind of analysis later. So we can collect it, which makes it usable. And then if you look at this this image on the left here, this kind of shows you that if you make ratios of different xenon isotopes, you can actually see, you know, was this emission from a nuclear power plant, was it from a nuclear explosion, or a medical isotope production facility. So just a real quick kind of blurb about the detection of xenon, how it works. So the detection works by looking at the near simultaneous emission of gamma and beta particles. And you have these two components. So you have a sodium iodide crystal and then a plastic scintillator. So crystal is the purple and the scintillator is the teal part. Those measure gamma rays and beta particles respectively, beta electrons. And those detections translate to emissions of light in the photomultiplier, which then turn into a count, which gets portrayed as a beta gamma spectrum on the next slide. So the detector spits out kind of this image you see over here on the left. And on the Y, you have the energy for gammas, and then the energy for betas on the X. And that kind of sets the foundation for analyzing xenon detections. So we have this spectrum, And this is a perfect lead-in to the current method, the current algorithm. It's called the ROI method, and that stands for region of interest method. The regions of interest are going to be these rectangles you see over here on the right, and they correspond to different xenon isotopes. So taking the example I put up for you here, the xenon 135, that translates perfectly to this orange rectangle over here. So the way the algorithm works is it takes this spectra emission on the left, and then it kind of puts rectangles on top of it. And if there are, data points, if there are emissions inside of those rectangles, the algorithm is then going to convert that to a count of xenon. So if we were to run this spectrum on the left through the algorithm, it would tell us that there's a lot of xenon 135. And then if other isotopes show up, it's going to tell us that as well. So what are some problems with this? Some significant issues are if the machine, the detector, is left out in the field for many years, we're going to see something called gain shift. And what this essentially amounts to is the axes on this spectra, they shift over time. They don't really stay perfect as the machine is kind of left to do its thing. And we see that the rectangles don't move, but the counts do. So on the previous slide, imagine if that spectra, kind of that streak for 135, imagine if that moved, either left or right down the axis, but the rectangles just stayed where they are. That means we're going to lose xenon counts, and it's not really going to give us an accurate, measure of whether or not there was 135 in the area. Another issue is overlapping isotopes. So I'll hit slide here to kind of zoom in for you. As you can see here, we have 131 metastable right here in this part. And then in this part, we have 133 metastable, but the 133 metastable peak actually leaks over into this region of interest. And as a result, we're going to get counts for 133M when it's actually not there. So that's a big issue. We kind of have this confusion amongst the isotopes. So bottom line, how are we going to fix this? We're trying to make an algorithm that's more flexible, can actually change and understand and read the different isotopes better than ROI. And that brings us to peak fitting. So peak fitting is a general shape fitting algorithm, and you take, just a spectra in the top left. Oh, there's a little echo. Spectra up here, and then you're using kind of shapes that already exist, kind of different probability distribution functions, and you're doing a 2D fit using kind of almost contour lines over the spectra to make it a little more unique rather than just slapping on a rectangle. So this is kind of a picture of how this actually plays out. So over here for the gamma, you have a curve. I'll talk a little bit more about the math on the next slide, but you have this curve, which is a Gaussian. And then over here for beta, you have the Fermi decay distribution. And you can actually combine these functions, combine the probability distributions to make a picture that looks like the spectra. So mathematically, how does that work? You have an amplitude here, A, which corresponds to each different isotope. It's kind of isotope dependent. And then the second one, for the beta emissions, you have the probability distribution function for that multiplied with the probability distribution function for gamma, which is going to be that Gaussian. So when you combine those two shapes that I kind of showed you on the previous slide, this one for gamma and this one for beta, you can then create a probability distribution function that corresponds to the whole spectra, the one I kind of showed you at the beginning when I was telling you, what the detector spits out. So you can just use, probability distribution functions to model the overall picture of what the isotope looks like. So now moving on to some error analysis. And why is this important? This is important because we want to see, does peak fitting actually outperform the ROI method? And if the answer is no, that's really problematic. If the answer is yes, that's exactly what we're intending to do. So error analysis is kind of going to allow us to do that side-by-side comparison with ROI and peak fitting. So the first step in this was doing Poisson statistics, and this essentially involved taking the result we get, this number, this xenon count, and taking the square root of that to give us the uncertainty or the error associated with our result. The problem with this was that the algorithm was essentially reading 0 wherever there was no xenon peak. So if there's no isotope peak, it read 0, which gives us the square root of 0. Wasn't super interesting, with regards to uncertainty. So we added in a background so that there's just kind of a consistent background count where there's not a xenon peak. And that helped us there have some more interesting results. And then additionally, the next step was obtaining error bars. And this is important because then if we plot peak fitting versus ROI, which you'll see later, we can actually do that comparison and see, you know, how they relate to each other. And this was obtained, we tried a number of different things. It kind of took a while, but we did a curve fit within Python to produce a covariance matrix. And the covariance matrix, as you can see below, it's a matrix that contains the covariance of different variables with each other. And interestingly enough, the diagonal terms contain the covariance of, you know, say the x variable with the x variable and the y variable with the y variable. And the off diagonal terms contain the covariance of, you know, x and y, y and x, kind of in that pattern. This is a general setup. And if you take the square root of these diagonal terms, it actually just gives you the variance, you know, of x and the variance of y. Or sorry, if you don't square root them, you get the variance. And then if you square root them, you get the standard deviation, you get your uncertainty. So the covariance matrix kind of was our starting point for getting the uncertainty for our fit parameters. And then we had to kind of do some manipulation to pick the ones that are relevant to the plots we were making. But this served as the basis for how we got our error bars. So once we got our error bars, it wasn't all smooth sailing from there, unfortunately, but we did get, you know, a matrix for concentration and then a corresponding matrix for uncertainty, but we still ran into problems where there would be a 0 number for concentration. It ended up giving us an infinity value, which we're still working through, because, you know, as you can see up here, this is just 131 metastable as an example. The algorithm read no 131 metastable present, but then because of this number, the covariance matrix and curve fit we were using just gave us, you know, an infinity value. It couldn't compute an uncertainty, but we still know if the algorithm gives us a 0 value, there's some error associated with the fact that it read no 131M. So we're still working through that, as I mentioned, and trying to kind of account for that. Another problem that we're also working through still is that the uncertainty, even when it does read an actual value, so say here and here, we're kind of looking at those as being significantly underestimated. And the reason is because we kind of missed some systematic error throughout the code. So now we're kind of going back in and saying, okay, what errors did we not account for? Because there has to be, you know, more throughout the code that we need to account for because it's underestimated. So we kind of pinpointed the problem there, and now it's just about going through the code and saying, okay, we need to add some error analysis here and make sure that we're propagating it all the way through. So now for the more interesting results. The results here are from a test, and this test involved running data that was already run through the ROI method, through the peak fitting method. So the data here contained 133 and 131M. The interesting thing there, I'll bring you back kind of to the overlapping isotope problem, is that 133 metastable in the ROI method was read as a false positive. So just to start out, the plots we created, this first one, concentration versus time for 131 metastable. Again, those error bars significantly underestimated, so you don't even see them on the plot, they're so small. But this is still really promising data because we see that ROI and peak fitting are at least in the same ballpark. You know, we don't have the error bars yet, but we see that they are doing relatively similar in performance. And then here is just the other isotope that was in the mix, 133. Same thing, error bars aren't showing up for this one because they're too small, but the data is kind of doing similarly here. So now back to that false positives issue. If you remember, you know, this isotope problem here, the bleeding into the 133, or sorry, the 133 metastable. If we go to the next slide, this plot kind of shows us this problem and how peak fitting is outperforming. So if we go to this red line here, this is called the minimal detectable concentration, the MDC. This line tells us that below the red line, those data points are kind of, you know, too low. The concentration is too low to really be detected. So they're almost ignored. We don't really accept them as, you know, actual counts of xenon. It's just kind of the machine misreading. And everything above the red line is valid. So what this plot is saying is that the ROI, this dark blue, actually read 133 metastable counts when there is none in the mixture because we were holding the cards, we knew what was in the mixture. But peak fitting, we don't see any of those points above the MDC, which is really awesome. And then two, in this plot, we see that the error bars are actually showing up, which is awesome, except for the infinity ones. But this plot is very exceptional for our results, and hopefully we're hoping to calculate an MDC for peak fitting, which will probably be similar, but it might have a little bit of a different MDC itself. So that's kind of our main result here. So just to summarize what I've talked about today, peak fitting is a new radioxenon analysis tool, and it aims to kind of solve some of the problems with the ROI method. My main contribution to this project was assessing the performance and kind of the error analysis. And then last, we plan to continue improving the error analysis and also to make some of the peak fitting algorithm components a little more robust. And with that, are there any questions?
Great job, Megan. So we're going to open the floor for any questions. We got a few minutes. I guess to get things started. So are the peaks, are they able to stretch or are they able to only to shift?
Yes, that's a really good question. So the cool part about peak fitting is that they do stretch and shift. It's a super flexible algorithm, and you can almost think about it as the peaks are kind of programmed to find the counts rather than the rectangles exist for the counts to fall into them. The peaks go and find, the peak fitting algorithm goes and finds where the xenon counts are.
Great.
Thank you, sir.
Hand raised. Go ahead, Lindsay.
Hi, Meg, thanks for your presentation. I really enjoyed it. One question I had, though, I was wondering what type of parameters go in. You talked about the MDC, I believe it was the minimal detectable concentration. What kind of parameters go into the MDC for peak fitting?
Yes, that's a really good question, and we have to do that soon, to calculate it for peak fitting. So the kind of general themes that you find in the MDC, it's this kind of long equation that's been found by a couple different national labs. So you have information about the isotope, about its specific decay. You have information about the detector, which is really important because that's where a lot of the problems with gain shift come from. And then you have sort of a unit conversion. So we'll be kind of finding that information for the isotopes and the detector moving forward.
Thank you.
Thanks, Lindsay.
Okay, well, other people are queuing up questions. Could you go back to your difference between the ROI and your, the difference between the two dots? Back a couple more. Yeah, okay, so I guess I'm curious whether, I guess the impact due to gain shift, how that would compare to like the differences you're measuring here between the two different methods.
So just to clarify your question, so kind of how gain shift plays out in this versus...
Right, because if you have gain shift, then it'll suggest a different concentration, maybe a lower concentration. And then so your technique to kind of do the same thing, just a completely different way. How does, I guess, the comparison or the differences between the two techniques shown here, how does that compare with, I don't know how gain shift changes over time or what impact or how big that impact is, but.
For sure. So the impact of gain shift kind of depends on largely on the detector. So we use a blend of real world data as well as data that we fabricated to kind of show us a certain thing. So gain shift would really be if you were doing a case study that was focused on a machine that was there and left in the field for a really long time. I'm actually not aware of the specifications for this specific test on if gain shift was a problem for the detector, but it will happen if you leave the machine out in the field. And I think you would probably just see that in kind of the difference between measurements for all the isotopes. So this test really honed in on seeing, are we going to see 133M false positives? It didn't look at, what's the general disparity between all the isotopes, but I would definitely assume that some of the differences between peak fitting and ROI in kind of these two plots would naturally come from gain shift. If that makes sense. I hope I answered your question.
No, it's good. Thanks, Meg. That's perfect. Does anyone else have any questions before we queue up our next speaker? Great. Well, good job. Yay. Okay, so we're on next. We're gonna a couple minutes to transition to Cadet First Class Lindsay Powley. She will be talking about excited state dynamics of DNA in 77K classes.
Hello, can everyone see my slides?
I can see, it's not presentation view, but I do see the slides.
All right.
Oh, there we go.
Okay, perfect. Hi, everyone. My name is C1C Lindsay Powley, and I'm here today to talk to you about the excited state dynamics of DNA in 77 Kelvin glasses. My mentor for this project was Dr. Kimberly De La Harpe, and we're both from the United States Air Force Academy. So what I'll talk to you about today, I'll go over the motivation for the reason why we decided to conduct this research, give you a little bit of background to help you better understand the research we'll be doing. I'll go a little bit in depth about the methods we use to collect our data and as well present some results and present our future work that we hope to finish before the end of the semester with this project and into the future as well. So a little bit of motivation on why I chose to do this project. Everybody knows that silicon-based electronics technology is approaching its limit. The conductors and metals that we use, semiconductors, the smaller we make them, they can only get so fast. So instead, we want to look on an even smaller scale to molecular electronics, which exploits the individual electric properties of single molecules. And we believe that DNA could be an optimal choice as a molecule to use in these molecular electronics because DNA is a designable, stable, and information-dense molecule that has the ability to readily self-assemble, and it's diverse and we can control its structure. Additionally, previous research determined that it's capable of charge transfer, so electrons can travel up and down the molecules. And so this is why we think it's a great molecule that could be used for molecular electronics, but in order to run, we first have to learn to crawl. So understanding the photophysical properties of DNA and DNA bases allows for us to assess its potential use for molecular electronics and other applications in the future. A little bit of background, a little bit of bio for all us physicists. What is DNA? We all know DNA stands for deoxyribonucleic acid, and it's a organic molecule that contains genetic code. It's primarily made of 4 DNA bases, guanine, cytosine, adenine, and thymine, which are linked to each other via sugar phosphate backbone. Can stack and form structures like base pairs, as you can see, single strands as well. Is very used to seeing when we a little bit more.
Lindsay, I think you're breaking up a little bit.
Of DNA
Could you possibly
scientists.
turn off your video feed? I'm sorry for interrupting.
Yes, I have to figure out how to do that because I am in video mode. Hold on, sorry. I'll turn off my video.
No worries, it's just breaking up just a little bit. Thank you.
Get back to where we were. All right, I believe this was the slide. Can everyone hear me now?
Yes, you're just fine.
Okay, perfect. So as I said, DNA, we can control its structure. Scientists can genetically modify DNA to fit our needs. There are different types of DNA and different secondary structures that it can form. So different types of double helices, the A, B, and Z form, as well as hairpin DNA, which is when a strand of DNA or RNA doubles back on itself and begins base pairing. And structures like G quadruplexes are also able to be formed. So for these reasons, we think DNA is a good choice for molecular electronics, and therefore it's worth it to examine its photophysical properties. So a little bit more about what we mean when we talk about photophysical properties. So when a photon hits a molecule and it excites it, we know that the molecule goes into a higher energy level and then relaxes back down to the ground state. So as it gets excited, we call absorption. It can go to a couple of different excited states. If it reaches a singlet excited state and fluoresces back down to the ground state without any intermediate steps, that's a rather quick process and it's called fluorescence. However, if something called intersystem crossing happens, the excited molecule can access a triplet excited state, which is spin forbidden, and therefore through quantum mechanics probability distributions, it takes much longer for those excited states to relax back down to the ground states. And those usually happen at lower wavelengths and take longer. That's called phosphorescence. And so with DNA, we're very lucky that it's a very photostable molecule. So that means if we get hit by harmful UV rays, our DNA right off the bat is not going to get mutated or change its structure because because it's being excited. That's why usually only older people get skin cancer, because DNA is so good at being photostable. And anytime a DNA molecule gets excited, it quickly relaxes back down to the ground state without any major changes happening. Unfortunately, that's bad for our research, because what we're trying to do is measure how long times of the excited states are. So because it happens so quickly at room temperature, what we want to do is go ahead and freeze our DNA using liquid nitrogen, where the 77 Kelvin comes in, because that effectively freezes out the vibrational motion of those molecules, which would help it relax quicker. So we have longer lived states that our instruments are able to detect. So here's a little bit more about photophysical characterization. Like I said, an excited molecule becomes excited through a photon, and it has a number of different pathways it can take, both radiative decays and non-radiative decays. So radiative decay are things like phosphorescence emission, but things like exomer formation, proton and electron transfer are all non-radiative decay processes. So we're looking to characterize those using spectroscopy. And just a little refresher, spectroscopy is a technique in which we shine a specific wavelength of light through a sample, and then look at the light that we receive after it's gone through this sample from a detector, and we're able to characterize different properties of that sample based on the wavelength of light we know we're shining at it and what our detector actually picks up. So in our research, we're using two different methods of spectroscopy. One's called transient absorption spectroscopy, which we use both an excitation source. In this case, it's a Q-switched neodymium YAG laser with a 3 to 5 nanosecond duration pulse at 10 hertz. And we use fourth harmonic generation to get a wavelength of 266 nanometers to hit our sample with, as well as a background white light xenon arc lamp provides that white light. So hitting our sample with both our excitation source and our probe source allows us to calculate the log of a difference between both the white light pump being on and the pump being off. And then we're able to calculate the change in absorbance in that molecule over a different, over a range of wavelengths to see at what wavelengths the molecule is absorbing. But the other method that we've been using primarily in our research is called time-correlated single photon counting spectroscopy, or TCS-PC. And this one's more restrictive, transient absorption, monitored both radiative and non-radiative processes. And this one hones in on the radiative processes. So we're using a pulsed laser to hit our sample at 90 degrees, and that causes those photons to excite the DNA molecules themselves. And once those DNA excited states relax back down to the ground state at some time later, those counts are picked up by a detector, both monochromator and a photomultiplier tube, in order to give us a distribution that looks like this. So it's based on a statistical approach. So those counts from the excited molecule itself are placed into bins based on the time they arrive at the detector. And once one of our bins reaches a maximum of 10,000 counts, the instrument stops. And from there, we're able to use a little bit of math and a little bit of data processing to back out the lifetimes of those excited states of our molecules. So just a little bit more about that. The top exponential decay function is the actual response that we get from the DNA itself when it's relaxing back down to the ground state. And the response itself, it almost looks like a delta function. That's our instrument response function. So there is a little bit that our instrument itself has to create. So those two functions are convoluted together into the output signal that we receive. So our job processing data is being able to back out what's part of our instrument response function and how much of that signal is actually from the decay of the molecule itself. That sounds like a lot of science. Here's how we actually did it in the lab. So we used a liquid nitrogen Dewar system, which consists of an NMR tube, which those top two pictures show what those tubes look like. So we prepared the solvent that we dissolved our DNA in. We had to try a couple of options to make sure that we wouldn't break the glass when we froze it, as you can see on the left side of the picture, until we settled upon a 10 molar lithium chloride solution, which we were able to dissolve our DNA in. So after we did that, we go ahead and dissolve our DNA in the solution. We froze our sample by simply placing the tube in liquid nitrogen Dewar system, which is basically just a holding tube that allows the sample to stay cold and submerged in liquid nitrogen throughout the instrument collecting data. And from there, like I said, we're able to perform analysis. So we used emission and absorbance spectra to characterize the steady state. And then we used our spectroscopy methods to look at more of the lifetime dynamic states of our nucleotides. So here's a little bit of our data, the nucleotide spectra that we were able to collect. So on the bottom, you can see the molecules that we use color-coded with their spectra on the graphs. So the absorbance spectra is on the left-hand side and the emission spectra is on the right-hand side. And it's interesting to note, right, our emission spectra takes place at 77 Kelvin because, like I mentioned earlier, DNA is so photostable that if we left it at room temperature with all that extra energy and vibrational, we wouldn't see these nice peaks. These peaks that are towards the left at shorter wavelengths, those are our quicker phosphorescence bands. And then on the right at longer wavelengths that are a little bit wider peaks, those are our, on the left is our fluorescence bands and on the right is our longer lived phosphorescence bands. And so after we ran TCSPC, this is the overlaid response functions that we were able to find, and using fitting functions, we were able to determine the lifetimes. So as you can see, the black curve itself, that's really, really shallow, really, really narrow. That's the instrument response function of about 1 nanosecond. And all the graphs, all the figures outside of that show that the DNA nucleotides that we were measuring do have longer wavelengths. When we look at the tables on the right-hand side, that is work done in 1966 by Isinger. He was the first really to do this technique, but instead of using a 10 molar lithium chloride solution, he used an ethylene glycol water solution. And so the slight differences in the results that we got versus the one Isinger reported may be due to the type of solvent being used. Additionally, our processing techniques allowed us to determine both a short-lived and long-lived component of the lifetime. So all those numbers in parentheses are our amplitude percentages of both the short-lived and long-lived time component. So for example, AMP has a 1.38 nanosecond short-lived component that dominates about 88% of our overall signal, while a longer 10.39 nanosecond component only dictated about 11% of our signal. So Isinger's results are presented as an average of both of those, whereas we were able to determine both individual components. And one thing interesting to note, what we have in red is CMP. We have 81% of it in our short-lived lifetime, which we resolved to be 0.44 nanoseconds. But since our instrument response function is approximately 1 nanosecond, we have reason to believe that we might need to use a different technique to more accurately capture the correct lifetime of these long-lived states. Of these excited states, and that could explain why Isinger in 1966 wasn't able to resolve a lifetime at all for CMP. So we've got all our nucleotides, just our little bases on how we did it, and we proved that it's consistent. Our next step was to collect and process data of more complex system. So we started with nucleotides, and then we moved on to single-strand polymers and duplexes, focusing mainly on adenine and thymine. So we started with AMP by itself, and then we did poly-A, which is a long string of A, as well as homo and alternating poly-A, poly-T strands. And so with this new research, these are what we decided to consider. Some questions we were trying to solve answers to is, are we seeing these same long-lived excited states lifetimes in polymers when we freeze them, as well as the nucleotides? Does it still work when we have a long chain, basically? Is there a difference in the impact between base pairing versus base stacking? So base pairing is when two nucleotides pair together, so A and T or C and G, versus just a bunch of A's on top of each other. And lastly, we were looking at how restricting our molecular motion overall impacts the dynamics of the DNA molecule itself. So this diagram from Kohler et al. Was a similar study to what we were doing. However, they measured DNA in solution. We're choosing to look at it in 77 Kelvin. So we want to see, are we going to see the same results in solution as we will see when we freeze our samples? And so a little bit more about this diagram. The important takeaways for here is in part A, that unstacked base has the quickest method to get back down to the ground state. So it's got the shortest lived lifetime. once you begin base stacking, it takes a few more intermediate steps to get back down to the ground level. So we expect to see longer lifetimes. And lastly, once we start pairing and stacking our data, we expect the lifetimes to be even longer then, because once again, there are more different pathways for that molecule, for the energy to take on its way back down to the ground state. And so we froze our DNA for our polymers, and then we looked at its steady state using both its room temperature absorbance spectra and its emission spectra while it's frozen. And again, you can see, just like with our nucleotides, we have our fluorescent bands, which happens at shorter wavelengths, and our phosphorescence bands, which happens at longer wavelengths and usually take longer. So here's our time correlated single photon counting response function that we got from the data, and then we're able to process to fit a function to find the lifetimes again. So in red, that's our smallest increment, basically just one nucleotide by itself. Black is our polyase, so just a single polymer. And then the blue curve is our duplex, so getting more complex as we move up. And what's very interesting to see is we can compare it to the previous work done by Kohler and Solution and find that our results are consistent. So the unstacked nucleotide has the shortest wavelength, and you can see because it's closest to where the instrument response function would be. Our stacked and unpaired polymer has a slightly longer wavelength than our nucleotide, but still it's not as long of a lifetime as our stacked and paired duplex, which is consistent and encouraging because in both Solution and in frozen glasses, we're able to see that we have consistent results. Some work we still need to do is to determine whether or not it's the base stacking or the base pairing that really contributes to that longer lifetimes. And that's going to take some more work because there are just so many options and things we can consider, basically uncontrollable variables right off the bat in our system, the way we measure this to determine definitively whether it's base stacking or base pairing that has the dominant component in creating those lifetimes. But just to summarize, in some future work, we looked at the steady state and dynamic photophysical properties of first nucleotides and then more complex system. And so in the future, characterizing it not only for AMP, but other nucleotides and eventually other sequences of DNA as well, will give us even more information about this. In our research presented today, we primarily looked at the fluorescence lifetimes of these molecules, but what's also interesting would be to characterize the phosphorescence lifetimes, and we're still working on resolving some of those. Additionally, in the future, we'd like to assess the dominant mechanism that determines the lifetime in these excited states. And we understand that photophysical analysis is an important first step to assessing the practicality of DNA use in future molecular electronics applications. Some of the references that I used to complete this project, and I was wondering if anyone had any questions.
Great job, Lindsay. Again, any questions you have?
Lindsay, I've got a question. It's kind of a naive one just on the basis of the work, but if you've got, if you're trying to make electronics made out of, molecular electronics made out of DNA, presumably you control the signals using currents and voltages as you do normal circuits. Why is how does the photophysics come in, the lifetimes of these things? How does that end up being important, the photophysical lifetimes?
Yes, thank you for the question, sir. Like I said, the photophysics is basically a first step to characterize, are we capable of really quantifying these radiative and non-radiative processes while we're doing it. I'll go back to this slide that kind of explains some of the different processes, but the photophysical properties of these molecules is basically like a first step. Like if the Apollo mission 11 the goal was to land on the moon, we wouldn't be able to land on the moon without having Vanguard first. So this is kind of like our Vanguard before the Apollo missions. But basically, we're looking at things, we want to be able to characterize how far an electron can travel down a strand of DNA. Well, in order to do that, we need to understand more about the different radiative decay and non-radiative decay processes that it can go through. So my diagram on the right, if you look in that red circle, things like exaplex and examer formation, I didn't go into detail on that, but other things like proton and electron transfer are all different aspects that we need to consider. And so in my last slide, when I talked about, oh, we can't definitively say whether it's base stacking or base pairing, that's because each individual molecule has these parameters to deal with. And when you start stacking molecules on top of molecules, there's a lot of interactions to go through. But so understanding one will help us eventually understand another. And ultimately, electron transfer is what we're looking forward to being able to definitively characterize, because we want electrons to travel up and down the molecule. That's how we're going to be able to get our signals and our changes in voltages and eventually electronics.
Okay, so maybe 30 seconds for this next question. Megan, do you have a quick one?
Yes, sir. You kind of hit on it in that question just then, but I know we want the lifetimes to be longer for molecular electronics, but can you just sum up, and you mentioned with like our skin absorbing UV rays and things like that, like why for molecular electronics is that the best outcome, like having the long lifetime?
Yeah, so the longer lifetime or at least ability to conduct that charge transfer is what's going to give us the ability to use long chains of frozen DNA as, conductors in the future. We're lucky biologically that our DNA is very photostable, because that means we're not walking around with mutated DNA from the day we're born, the day we're exposed to the sun. However, that also works against us because it happens so fast when we want to use a scientific application, use DNA as a molecule for other than biological purposes. Does that answer your question?
Yes, it does. Thank you, Lindsay. Awesome presentation.
Thank you.
Awesome presentation. Great job, Lindsay. So next we have C1C, Jacob DeLange. Feel free to go ahead and share your screen.
All right,
And
thank you, Major
his talk
O'Keefe.
will be on spectroscopic analysis of radioactive xenon. I see your slides. There we go. It's good.
Perfect. All right. Good afternoon, everyone. I'm C1C Jacob DeLange, senior at the Air Force Academy, and I'll be discussing the spectroscopic analysis of radioactive xenon. First of all, I want to thank all the members of our project, my research mentors, Dr. Michael Schaefer, Captain Sarah Castro, and Dr. Randy Nyes. And also a big thanks to Cadet Dan Reinford, who's been working on the project with me. So a little bit of overview of what I'll be talking about today. First, I'm going to introduce and provide the motivation for our project. Then I'll walk through the background and theory that underpins it. Then I'll lay out our experimental approach, and then finally I'll wrap up with some conclusions and next steps. So starting with motivation, xenon is extremely useful in nuclear treaty monitoring for 3 reasons, and Meg already hit on these, but I'll just go over them again. First, xenon is a common fission byproduct, so it's produced in abundance during a nuclear test. Second, its radioisotopes have low background levels, so they stick out like a sore thumb whenever they are created. And third, because xenon is an inert noble gas, it's difficult to contain and therefore easy to detect remotely. Now, there are existing systems for remote xenon detection, most notably the SANA system pictured below, but these utilize methods such as coincidence counting and mass spectrometry, which are limited both in their precision and their selectivity. For example, coincidence counting can't pick out xenon's stable isotopes because they don't undergo any sort of nuclear decay. And mass spectrometry struggles to distinguish between xenon's isotopes and their corresponding isomers because they're so similar in mass. So to overcome these obstacles, the Laser and Optics Research Center here at the Air Force Academy is developing an atom trap trace analysis, or ATA system, that overcomes these barriers in selectivity and also offers increased sensitivities. In fact, we estimate that for every 10 to the 16 atoms of background gas, we can pick out 1 atom of interest. And the way our system is able to do this is because it utilizes laser cooling and trapping. And the way that works, if you look down at the energy level diagram, we start by exciting our xenon into a long-lasting electronic metastable state, which has a lifetime of about 30 seconds. And then from there, we use an 882 nanometer infrared laser to drive it on this cycling transition. So we pump the xenon up to the cycling state, which has a lifetime on the order of nanoseconds, to almost immediately decay back down to the metastable state, and only the metastable state. And in so doing, the xenon will lose a little bit of its momentum and be cooled. And because the metastable state is so long-lasting, we can drive this transition hundreds or even thousands of times until the xenon is sufficiently cool that we can trap it. And the other nice thing about the cycling transition is it's actually unique to each one of xenon's isotopes. But the only problem is we don't yet know these values for some of xenon's radioisotopes, most notably the ones shown on the screen. So the ongoing goal of our project is to determine these values experimentally. So getting a little deeper into the theory, there are two effects which account for the changes in cycling frequency between xenon's isotopes. The first is the isotope isomer shift, which comes from changes in nuclear mass and nuclear charge distribution. And the second is hyperfine splitting, which comes from the coupling of your electronic angular momentum with your nuclear spin. So if you look down at the figure, I can give a little visual intuition for that. We start at some reference in frequency space, which we treat as a 0 point. And then your isotope isomer shift moves you over to the center of gravity of your hyperfine spectrum. Now that doesn't actually correspond to a hyperfine peak, but it is the point in frequency space from which all of your hyperfine splittings are measured. So if you look, each one of these short little blue lines corresponds to a different hyperfine peak in this arbitrary spectrum. You can see they're all measured with respect to the center of gravity, which itself is measured with respect to the reference. Moving a little deeper into hyperfine splitting, I have the equation that governs that shown here. It's a little bit of a monster, so let me walk you through the important parts. First, you'll notice the quantum numbers I, J, and F. I corresponds to your nuclear spin, J corresponds to your electronic angular momentum, and F is the magnitude of the vector sum of I and J. And because F is defined in terms of a vector sum, it can actually take on many different values. So if you look down at the table, you'll see each isotope has many different possible F values, each one corresponding to a different hyperfine splitting. And the other big thing to take away from this are the A and B hyperfine coefficients, which tell you the magnitude of your hyperfine splitting and come from different nuclear parameters. And I'll talk a little bit more about those in a few slides. So here's a little bit more visual representation of hyperfine splitting. You can see that each one of our each one of our states of interest, the metastable state and the cycling state, split into multiple different hyperfine levels. This allows for many different possible hyperfine transitions, but only one of them, shown in red, is the cycling transition we're actually interested in. Now, there is also the loss transition, which goes to the hyperfine level just below that of the cycling transition. Now, if the loss transition is sufficiently close in frequency to the cycling transition, there's a chance that we could accidentally drive the xenon on this transition, and it'll fall out of resonance with our laser, and it will no longer be able to be trapped. So we want to be aware of this because there are a few tricks we can play if that is the case. So previously, we've actually done a theoretical estimation of the hyperfine spectra of our radioisotopes of interest. So starting with the isotope shifts, as it turns out, these vary linearly with mass numbers. So if you look down at the figure, you actually get two different regressions for your odd and your even isotopes based on their nuclear mass number. And so using the data for xenon stable isotopes, where the isotope shifts were known, we were able to figure out those values for the radioisotopes of interest. Now, for isomers, that was a little trickier because there's not a lot of data on their nuclear charge distributions. So what we did is we looked at cesium, first of all, because it's close to xenon on the periodic table, and second of all, because its isomer shifts had actually been measured. And what we did is we looked at the maximum difference between the shift for an isomer and the shift for its corresponding isotope. And we found that value to be about 450 megahertz. And we interpreted that as a range of error on our center of gravity. So if you look down at the figure in the spectrum for xenon 133M, a red dot corresponds to the center of gravity. And you can see there's a range of error through which that center of gravity and the rest of the hyperfine spectrum along with it can shift. And finally, for hyperfine splitting, we had to estimate the hyperfine coefficients. As it turns out, A is proportional to your magnetic dipole moment divided by your nuclear spin, and B is proportional to your nuclear electric quadrupole moment. And so using these values, which are known for xenon's radioisotopes, and then some of the hyperfine coefficient values known for xenon stable isotopes, we were able to extrapolate or estimate the values of the hyperfine coefficients for xenon's radioisotopes. So next, moving into our experimental approach, there were 5 main components which we needed to address. First, how we're going to acquire and handle our samples. Second, the viability of our experiment. Third, how we're going to input, circulate, and remove our radioxenon from our system. Fourth, how we're going to scan the frequency of our laser. And then finally, how we're going to acquire and process our data. So starting with sample acquisition and handling, we've identified Idaho National Labs as a potential source for our radioisotopes. The thing is, because these are radioactive gases, we can only store a certain amount of them at a time, limited by the sample activity. So for most of the isotopes, we can only store about 100 microcuries, but for xenon 131M, we can actually bump that up to about 1 millicurie. And then the other big consideration here is whether or not the radioisotopes are going to come in separate containers or whether they'll all come mixed together. So they do come mixed, we're going to need to devise a method to differentiate them from each other when we run our actual experiment. So moving on to the viability, because we're working with such small samples, we wanted to make sure that our system would actually be able to detect them. So what we did is we considered Xenon 135, which has the shortest half-life of about 9.14 hours. And so given our sample activity of about 100 microcuries, that limits our sample size to about 1.73 times 10 to the 11 atoms, or a volume of about 7 picoliters at STP, which is not a lot. And so using our estimated flow rate and trapping efficiency for our system, which we got from experiments with stable xenon, we estimated that we'd be able to trap less than 1 atom per second of Xenon 135. That's not a lot, but the thing is, when our xenon is actually trapped, it's still being driven on that cycling frequency and will continue to fluoresce at that 882 nanometer wavelength, thousands or even millions of times per second. And so we can actually measure that fluorescence. And so even for this small number of trapped atoms, we'll be able to pick that out, we believe. But on top of this, we've also implemented a few improvements to our system to improve our atom trapping rate. And the main thing is we've implemented a more powerful laser. It has about 3 times the power of our original laser, and that should dramatically increase our trapping efficiency. And so we should see that the count of atoms per second increase. Moving on to our vacuum system, for our input and removal method, we'll be using activated charcoal, which is a substance that at low temperatures contains xenon, but at high temperatures, about 300 Celsius, it'll release it. And that gives us the ability to contain or release our xenon at will. In addition to that, our larger vacuum system needed to fulfill 5 functions. First, it needed to be able to load our sample. Second, it needed to be able to prepare it by mixing it with argon, which is just a step that helps some of the other components of our systems run more smoothly. Third, we need an open mode, which is where when we're done with our xenon, we can just dump it into the atmosphere. Obviously, we won't be doing that with our radioisotopes, but we do also do experiments with stable xenon where we need this open mode. We also need a recirculation mode. That's what we will be using with our radioisotopes to make sure they don't get released into the atmosphere. And then finally, we need a way to recollect our radioxenon samples when we're done. Moving on to laser scanning. The reason this is important is if we come across some peak that corresponds to a cycling frequency, we need to be able to pinpoint it at a specific location and frequency in order for that information to be useful. And so the way we need to, the 2 things we need to do that are first, an atomic reference which we can treat as a 0 point, and second, a way to scan the frequency of our laser relative to that 0 point. And as for the second method, or for the first, we'll be using Xenon 134, whose cycling frequency is known. And for the second, we'll be using an electro-optical modulator, or EOM. What that does, if you look down at the diagram, it takes some carrier signal shown here in the middle, and it puts sidebands on it by rapidly modulating the phase of that carrier signal, where the frequency difference between a sideband and the carrier signal corresponds to the frequency at which you're modulating the phase of the carrier signal. And so in order to scan our laser, what we're going to do is we're going to lock one of the sidebands. In this case, you can see the leftmost sideband is locked to the peak for Xenon 134. And when it is locked, it'll stay in place. But as we change the modulation frequency of our EOM, the carrier signal and the other sideband will move with respect to that leftmost sideband. The only problem is in a cell of atmospheric xenon, which is what we've used in previous experiments, you have multiple different peaks which can be locked to. So as you're scanning this other sideband, there's a chance that it could lock to another peak. In this case, Xenon 136, in which case your original lock will break and you'll jump locations in frequency space unexpectedly, which will ruin your data. So to work around that, what we're doing is we're just going to use a single isotope cell, which only contains Xenon 134. Now, another thing to note is that in our magneto-optical trap, where we'll actually be scanning our laser over our radioxenon spectrum, we will not be modulating that signal. So only the carrier signal will be present in that spectrum, whereas inside of our single isotope reference, you'll get the whole setup with the sidebands. And then when we actually run our experiment, what it will actually look like is this, where like I said, the leftmost sideband stays put while the carrier signal and the rightmost sideband scan. And you'll also notice the carrier signal in the radioxenon spectrum and the carrier signal in the single isotope reference scan in parallel to each other. We'll always know where our laser is at and always be able to pinpoint those peaks that correspond to cycling transitions at a given frequency. And then finally, for our data acquisition and handling, we'll be pulling data from 4 different sources. First, a camera, which we'll use to measure the fluorescence of our trapped xenon. Second, the RF generator, which drives our EOM, which tells us our frequency relative to our lock. Third, a wave meter, which tells us our absolute frequency. We just use that to monitor and check the results from our RF generator. And then finally, a series of photodiodes, which we use to monitor our laser's power. And then here I have a logic diagram displayed for how we're going to be acquiring our data. The highlights from that are, first of all, we will not be taking all the data from our camera's field of view. Before we start our experiment, we're going to run a test with some stable xenon and figure out the region in the camera's field of view where the trapped xenon is showing up. And we'll only be pulling the data off from that region to try and eliminate as much background noise as possible. And then to further limit that noise, we'll regularly take images of our background and we'll subtract those from subsequent images to try and negate the effects of hot pixels and also scattered light in the background. And then in conclusion, I have some next steps shown here. We've actually made some progress on these since I made the presentation. We have actually implemented our scanning method. Our EOM is installed, and we've checked some of its limitations. We've figured out how to interface with the majority of our measurement devices, and we've written the bulk of our data processing code. The only thing left for us to do is actually to acquire our radioisotope samples. But the one thing we want to do before we do that is run our experiment with a pseudo sample of stable xenon. This is just to troubleshoot our system, make sure our vacuum is good to go, our data acquisition process works, our laser scans the way we want it to. Once we've done that and we've worked out any kinks, we'll be ready to actually get our radio xenon samples and run our experiment for real. And that concludes my presentation. Here are my references. Are there any questions?
Thanks, Jacob, for the talk. Does anyone have any questions for him? Go ahead, Zachary.
Hey Jake, great presentation. How close to those isotope peaks do you expect to have to be in frequency space before you actually see a response from the machine? And then how soon after reaching some point in frequency space do you think the machine will actually respond to that peak, if that makes sense?
I think you're muted, Jacob, if you're explaining. You're still muted. Hello?
Maybe he just didn't like my question. I don't know.
I'll see if I could unmute him. I don't know.
Yeah, sorry about that. My computer froze on the presentation screen for a second. So if I go back to this slide here, I'm showing the peaks as just straight lines. They're not infinitely thin lines. They have a line width. Comes out to about 5 megahertz. So when we're getting down to within a megahertz or two of that peak, you'll start seeing a response, which is why for our EOM, our scan rate, we're going to be scanning about 1 megahertz every second. Does that answer your question, Zach?
Yeah, thank you.
Perfect.
Jake, I've got a question. When you're scanning, do you know if you've reached a peak if you see fluorescence and basically you start to trap atoms?
Yes, basically. So that's kind of why we need to make sure we eliminate the effects of noise, because there is going to be scattered light at all times and our camera will be picking that up. So we need to make sure that we don't register that as a peak. We need to establish that as a background level and only treat any like large spikes above that background level as atoms being trapped.
And if you're trapping 1 atom per second, I mean, that's quite small. You anticipate you'll be able to see a peak in the 135, for instance?
We do, yeah. Like I was saying, if I go back here, that 1 atom that's trapped at a time will fluoresce thousands or even millions of times per second. So that's thousands of millions of photons, and that's what we'll actually be counting.
Okay.
And so that will correspond. It won't be a large spike, but it'll be large enough that it'll stick out above that background level.
Do you know when you're getting your samples in?
We don't have a timeline for that yet. We're wrapping up on our vacuum system right now, but after that, we should be ready to start doing dry runs.
Okay. Thanks.
Jake, are you gonna be able to continue this when you graduate?
Maybe. I don't know. Maybe if I go to AFTAC, maybe they'll do something with this.
Anyone else have any questions for Jake? Great. Good job, Jacob.
Thank you, sir.
Now gives us plenty of time to transition to our next speaker, C1C Nicolette Clark. So her talk will be on machine learning for 2D modeling of space weather. Space weather.
My computer's not cooperating perfectly, so I'm just going to go ahead and share my whole desktop. Major O'Keefe, would you let me know if my slides are showing up?
Not yet. Not yet. Doo-doo. You might
Still
have to
nothing?
act. Nope, still nothing. You might have to act it out. Do some demos.
I think I'd have a difficult time doing that one, sir.
Right. I think I have possibly an older version of your slide.
How about now, sir?
Negative. Oh, there we go. There we go. Yes, yes, we're good.
All right,
You're
cool. All right, sounds good, sir. And would you let me know if I start to break up so I can turn off my video?
Right. Yep. You're good right now, though.
Sounds good. Hi, everyone. I am C1C Nikki Clark, and I'm with the US Air Force Academy, and I'm going to talk about my research in machine learning techniques for two-dimensional modeling of space weather. This is a quick overview of what I'll be talking about. So I'm going to start out with where this project came from and why we're doing it. I'll go through our data set and what we did with it, and then I'll talk about the results of the machine learning techniques that my group used, and then I'll compare them to a different model called International Reference Ionosphere, and then I'll just briefly touch on future directions that this work could take. So this project started some time ago, a number of years ago, with an instrument that the physics department created called the Integrated Miniaturized Electrostatic Analyzer. And this was designed to be a low power and low mass instrument that could be mounted on a number of different host satellites that could take in situ measurements of plasma density in the ionosphere, with the long-term goal of using the data that the IMESA instrument collected to create a real-time map of space weather. So the approach that my group took to this project was to take that IMESA data set that we have and apply machine learning techniques to it in order to try to create a model of the plasma densities in the ionosphere. The purpose for this is to give satellite operators a tool to be able to see what types of conditions their satellites are facing in real time, because space weather can cause some different issues with satellites, such as in the case of where you have variations in plasma density that can cause scintillation and breaking up of the radio waves that are used to communicate with the satellite. So, if you're using a satellite for a mission essential operation, you would like to know if you're going to lose contact with it because of space weather. So having a real-time map of this space weather will help satellite operators be able to see when that's coming and make contingency plans to still accomplish a mission, even if there's space weather issues. I'm not going to go deeply into the network, but I just want to give kind of an overview of what a neural network is and what it does. So what you can see right here, I'm just going to laser pointer out. What you can see right here is that we have two different types of nodes, which are represented in green squares and blue circles. These green squares are data nodes, which on the left are the inputs, which are numerical data values that we give to the neural network on which it bases its predictions. And then over here on the right, at the end of its computations, the neural network will give us an output, which is the predicted value of density, plasma density, for the particular time and location at which we wanted it. In the middle are what we call hidden layers, and these blue circles are computational nodes in which the network is actually performing a linear computation. And we call these hidden layers because we don't see the intermediate outputs of each individual computational node or neuron. So we don't see those, hence hidden layers. And inside each node, as I said a minute ago, there is a linear model, and all of these linear models working together are able to identify underlying patterns in the data set and essentially map out how the inputs that we give to the model are related to that output of plasma density. So we used 815,000 training points and 204,000 testing points, which means that for 800,000 of the data points, we gave the model the inputs and the associated output that was measured by IMESA. and it used those data points to map out the relationship between the inputs and outputs. And then 204,000 testing points, we gave it just the inputs and asked it to predict, and then compared those predictions to the actual IMESA measured densities. So the inputs that we gave it with which to predict plasma densities were the location of the satellite at the time of measurement, so the latitude, longitude, and altitude, also the solar elevation angle, which is how directly overhead the sun is. We also used these three, what I'm calling magnetic indices. It's a little bit of an abuse of the term, but I'm just using it to be as succinct as possible. IMF is interplanetary magnetic field, which is the sun's magnetic field. F10.7 is solar radio flux at 10.7 centimeters, which encapsulates information about solar activity, and KP is a geomagnetic index that is used to determine it contains information about the disturbance in the Earth's magnetic field. So those were the inputs that we gave to the model on which to base its predictions. We also had to do some curation of our data sets. The first thing we had to do was to the IMESA data set, actually. This data set contained a lot of entries that were labeled as NaN, which means not a number, and essentially that is an error message that the IMESA instrument produces when it is not able to measure the plasma density because the density is too low. So that instrument can only measure down to a threshold value, and that is one of the limitations of this data set, is that we are missing some values because the instrument could not measure the density because it was too low at that point. And then in order to synchronize the data from multiple sources, those multiple sources were the IMESA instrument data set itself, and then also these magnetic index measurements that I talked about a minute ago. We got those from an online database called OmniWeb. So to synchronize them, we had to do some interpolation in order to get the magnetic indices at the times that we wanted. Those times were the same time that each IMESA density measurement was taken and 24 hours into the past. So essentially for each IMESA density, we had one measurement of IMF, KP, and F10.7 at the time the measurement was taken. And then we had also those three magnetic indices at hourly intervals over the past day. So there are 25 inputs for each of those three indices. And then there were 5 inputs from the other things I talked about, the location and solar elevation angle, for a total of 80 inputs per density measurement. What you see here in this picture is one result of a plasma density distribution that was predicted by our model. And what we did to create this is we gave the model a set of inputs in which we held constant all of the time-dependent inputs. So we held constant the IMF, KP, and F10.7, and then we varied the latitude and longitude over their whole range. So essentially we were saying to the model, at one point in time, predict the plasma density everywhere over the globe. So you can think of this as a snapshot of our results at one point in time. The way that we measured the network's performance was using correlation coefficient, and the R value that we got using a cross-validation approach was 0.556. If you've used correlation coefficient to evaluate performance before, you are probably scratching your head because this is a fairly low R value. And yes, it is pretty low. And the reason for that is we are using a very, very sparse data set. So one analogy I've used before is imagine that you were to go outside and take a measurement of the temperature right outside your back door here in Colorado Springs, and you tried to use that one measurement to create predictions for the temperature everywhere across Colorado, the whole state. You would not expect to get a very accurate result from that because you're using very, very little data in order to try to predict a lot of data points. And that's kind of the problem we were facing here. We had very few data points on which we were trying to base our model. We're trying to take a set of data, about 1 million points collected over 5 years, and predict the plasma densities essentially everywhere in space at whatever time we want. So it's a lot to ask of the model. But we've got a paper by someone named Lehmann who said that for a model of geomagnetic parameters such as this one, if you see an R value of 0.5 or higher, that's actually pretty good given what you're asking the model to do and the scarcity of data. And so we were pretty pleased with that R value of 0.556. Another thing I want to briefly mention, yes, I know these plots are really small. This is just a quick look at the data set that we were actually using. This is just the IMESA data points just plopped onto world maps based on the season they were taken in and the year they were taken in. The only thing I want you to look at with this is how inconsistent our data measurements actually were. So as you can see, in some seasons like the spring of 2014, we had almost no data at all, whereas other times we had over 100,000 data points. Sometimes we had more measurements in the northern hemisphere and sometimes in the southern hemisphere. And the reason I bring this up is, in addition to the sparseness of our data, we were also having some issues with the data set actually being kind of spotty based on when those measurements were taken and where. So this data set was a little bit inconsistent. And so working with that and the fact that we had very sparse data, we were very pleased to get an R value of 0.556. We also evaluated our model's performance during times of geomagnetic storm and geomagnetic quiet. And we used a time period from 16 to 26 March because at that time there was a geomagnetic storm directly followed by a period of quiet geomagnetic activity. And the reason I show you these plots is to talk about how we selected stormy versus quiet times. So we based that on the KP geomagnetic index. And when KP was elevated above about 4, we considered that to be a geomagnetic storm. And when it was below 4, that was considered a period of geomagnetic quiet, which is important to our results that I'm going to show you next. What you see here is 2 different things plotted on the same graph here. In black, we have the actual densities that were measured by the IMESA instrument. And in red, we have the densities that were predicted for those exact same times and locations by our model. And what you see here is in this upper plot, during the stormy period of time, our model was not predicting the full spread of data points, but it was predicting a wider spread than it did during the period of geomagnetic quiet. So it didn't do a very good job at all in this period of geomagnetic quiet. Didn't capture those peaks in the density. And so from this, we were able to infer that our model is actually placing too much emphasis on KP, which means that when KP is elevated, such as during the stormy time, then the model makes better predictions. It predicts a wider spread of plasma densities, and in times of lower KP, it does not do as well. It does not predict that the densities will be as high. Next, we compared to International Reference Ionosphere, which is considered to be a internationally recognized standard model for predictions of the ionosphere. It is another model, so this is not truth data, this is not measured data points, but it is considered to be one of the better models out there. This top plot is a plot of the IRI predictions on 24 March at 1700 UTC, and the bottom plot is for the same time period, the predictions of our model. So there's 2 things that you can see where our model is not agreeing with IRI. And the first is in the magnitudes of the densities. Our model is predicting lower densities than IRI is. And this has to do, we believe, with the problem that I talked about on the previous page, where we are not seeing our model predicting the full spread of densities that were actually measured. Additionally, the other thing that you'll probably notice right away is the model did not predict the same resolution of spatial features that IRI does. So in IRI, you can see the separation between crest of the Appleton anomaly, and our model does not show that. And we think the reason for this is insufficient data, because we just do not have enough data over time to resolve those different spatial features. Lastly, we did a quantitative comparison to IRI. So what we did is over the period of stormiest geomagnetic activity and then the period of quietest geomagnetic activity, we created at hourly intervals pairs of IRI and model predictions. So just like I showed on this previous slide, we have here a pair of IRI and model predictions that are matched at the same time. We created these types of distributions at hourly intervals over the stormiest day and the quietest day. And then we compared those pairs of distributions using the correlation coefficient and also the root mean square error. And on the left here is the plot of correlation coefficient. And what you can see is that for the stormy day, especially towards the end of the day, the model's correlation to IRI, how well it compared to IRI, increased significantly. And We think that one of the reasons for this is the model's better predictions during stormy days. And that can also be seen on the root mean square error plot. On the stormy day, which is the red line, we had generally lower root mean square error than we did during the quiet day. We also noticed a trend that I mentioned a moment ago of better performance towards the end of the day on the stormy day. And this may be a localized trend. We haven't analyzed this type of plot over a long enough time period to know whether or not that was just localized to this particular day or not. So that would be another way we could take this. One last thing I wanted to mention about the root mean square error. That's kind of a misleading sounding term. Root mean square error here refers to the difference between IRI and the model, not an actual error in the model, because as I mentioned, IRI is just another model. It's not truth data. Some future directions we could take this work would be to try to improve the dynamic range of our model by probably changing the loss function is the first thing that we would try. And also we could compare our model's performance to IRI over a longer period of time. Are there any questions?
Great job, Nicolette. Do we have any questions?
Yeah, Nikki, this is Dr. Chun.
Yes, sir.
Hey, if you go back to slide 10, I know that you said that your model, your machine learning model, was underperforming in terms of comparison to IRI, but really it wasn't that much. You're kind of basically at the same order of magnitude. You're just a factor of maybe 2 off.
Not 2, yes, sir.
So I wouldn't put down your model map,
Yes, sir.
too much.
So when I was talking about this comparison, the reason I mentioned that the machine learning model is making lower predictions is because that actually is related to the result that we saw elsewhere, like in this plot where we're comparing to actual predicted values. So that was one possible reason that we thought we might be seeing somewhat lower values here. We also did find some literature on IRI that said certain formulations of IRI can tend to run a bit high. So it could be that also.
Yeah, thanks.
Yes, sir.
Let's see, I guess I'll ask another question. Oh, go ahead, Colonel Scoville.
Nikki, is there sufficient resolution in your data, the black and red spikies, to see the dip in if a satellite was crossing that IRI model where there's the bright band and then it's cool and then bright again, I'd expect the density to go up and then quickly down and then up again as it passed through those bands. Do you have sufficient resolution to see that in the data here? Or?
That's a really good question there. And this is actually not the best plot I have in here for looking at that. That's one of the things that we've been looking into more kind of since I put this together. But this first plot here is actually something where you can see that a little bit. And the answer is we really don't have good enough resolution for it. What you see here in this plot is we binned all of our IMESA measurements by their location, and then we average them. So essentially this plot shows the average density that was measured by the IMESA over a 5-year time period at each location. And that's why the maximum here is kind of smeared out, because this is average across time. But what you'll notice is in this average plot, we really can't see that separation between the two crests, except maybe possibly around Southeast Asia. But the short answer to your question is we don't really have that resolution in the data set.
Okay. Yeah, I would, definitely wouldn't be surprised to not see those bands then on your model if the data set doesn't even have the resolution to show it. Thanks.
Yes, sir.
Dr. Giblin, sir, do you have a question?
Yeah. So thank you. Nikki, good job. Really good presentation. Well, what exactly is the time resolution of your data again?
So you mean the IMESA data set, sir?
Yes.
Yes, so that's kind of a complicated question to ask to answer because it has a lot of gaps in it. So technically the IMESA instrument takes a measurement once every approximately 10 seconds. But in a data set of initially 8.5 million data points, 7.5 million of those were recorded as errors because the IMESA instrument couldn't measure the plasma density, in most cases because it was too low. So we do actually have large gaps in the data set, meaning that there is not that doesn't stay constant. There are varying time gaps between data points.
Okay.
Does that answer the question here?
Yeah, and so roughly how many orbits worth of training data did you use roughly? Do you know that?
So not off the top of my head, we used 5 years worth of data total, and the orbit period is just over 90 minutes for the satellite. So the difficulty is that I don't know exactly how many orbits worth of data we had to remove because it was recorded as an error message.
Okay, got it. Nice work. Really enjoyed the talk.
Thank you, sir.
Thank you for your question. Thanks for your presentation, Nikki. Good job. So next we have C1C Zachary Eiler on building a dye laser using an optical nanofiber. So Zachary, go ahead and share your screen. Oh, nice. I see it. It's good. And you are ready to go.
I had to unmute myself there.
We can hear you now.
Okay, sweet. So good afternoon. My name is Zach Eiler, and I will be talking to you about building a dye laser using an optical nanofiber. So I want to start out with a quick overview, and I'm going to have this presentation basically in the form of questions that someone might have when they hear about this project. So what is an optical nanofiber? Why are they interesting? How do we fabricate one? And how do we use this to build a laser? What did we achieve so far? And what are our plans for future work? So what is an optical nanofiber? So an optical fiber is basically just heated and stretched to form a taper region. So as you stretch it apart, the middle section gets thinner, and there's some special ways we do that stretching so it's predictable. And then this taper region consists of a linear and an exponential portion. So we start at this about 125 micrometer diameter, which is the core and cladding of an optical fiber. This goes down linearly with our taper, and then finally it reaches about 6 micrometers, and then we enter this exponential portion where we really can't control the linear taper anymore, so we just stretch it out, and that gets us down to about 1 micrometer. So again, we're taking that about 125 micrometer diameter optical fiber to start, and at the end, this gets down to about 1 micrometer. Ideally, we would be below this 1 micrometer level. I'll talk about our results from that in later slides. So what is an optical nanofiber laser? So here you can see the core and cladding of the optical nanofiber begins at about 125 micrometers. This tapers down all the way until we reach our minimum diameter, and then it tapers back up again, back to a normal optical fiber diameter. And the idea here is that for these small diameter optical nanofibers, there's a strong evanescent wave that extends into the region outside of the fiber, and that actually interacts with, in our case, a gain medium, or it can just interact with the environment around it. Why are we doing this? So nano, a lot of applications in multiple areas of physics. So you can create novel laser designs. For us, we're creating this nanofiber dye lasers, but there are also alkali lasers that you can create. They can be used to create optical sensors, robust and highly sensitive, and you can use them to sense a variety of different things, including force, displacement, temperature, and even pH. And then finally, there's atom trapping. So if you launch red and blue detuned light into a optical nanofiber, the evanescent wave from each of these different lights creates an attractive and repulsive effect, which can actually be used to trap atoms. And that can be useful in quantum information science, where you're actually trying to manipulate these single atoms that are trapped by the optical nanofiber. And right now with this project, we're really trying to get experience fabricating and handling these optical nanofibers. So they're incredibly difficult to handle, as you could probably assume, with diameters that are in the 1 micrometer range. And so that's kind of the goal of this experiment, in addition to building the working dye laser. So optical nanofiber pulling. I'll go through the setup really quick. It's based on setup by Hoffman in 2014. We essentially have these 2 linear stages set up in multi-axis mode so that they can move at variable speeds. In the middle, we have this flame nozzle that uses a hydrogen-oxygen flame to actually heat the fiber as we stretch it. The entire time we're monitoring the fiber through 2 microscopes just to see how the pull is going, and if anything breaks mid-pull, that will ruin the fiber. So how the pulling actually works is we have both stages moving in the same direction. That's indicated by these arrows here. It's just that one stage is moving slightly faster, and this creates a much more predictable and slow pull than if the 2 stages moved in opposite directions. And conservation of volume is what is used to predict the nanofiber parameters after pull, and it's used to define basically steps for these linear stages to take in order to create a nanofiber with a desired taper region. The alignment and cleaning of this step at this step is critical. If you have any dust or dirt or any of the buffer that covers the glass of the original nanofiber or optical fiber, if that's left on the region that is affected by the flame nozzle, that can cause burns in the optical fiber or just to break altogether. And if any of that happens, you basically have a nanofiber that is useless. And then once the pull is actually done, we leave the fiber here and create the optical cavity around the fiber. So we try not to handle the optical nanofiber at all once it's created, just because it's so difficult to do that. For the nanofiber pulling results, the current nanofiber diameter is below 10 microns. So here's a profile of one of our more recent pulls. So we start out at this 125 micron level. We start tapering down linearly, and then we enter this exponential region down to our edge finding routine says we're at about 7 micron, but we are approaching the microscope resolution and edge finding technique limitations. So this is an old nanofiber that we pulled. The program indicated we were about 8 microns. This is our new fiber, and the program indicates we're about 7 microns, but by my measure, we're at least half the diameter of that old nanofiber. And you can definitely see that the resolution of our image is very low. So it's very hard to actually find where the edges of this fiber are. And so that's kind of the limitations that we're approaching. And a newly compiled pulling algorithm might be able to push our fiber diameter into the single micron range. That's yet to be seen, though. We're addressing other problems with our setup right now. And we think our fiber diameter should be good enough that we'll be able to create a laser. So a dye laser has already been created using tapered optical fibers. This was done by Payne in 1993 initially, but there have been a lot of results since then. So some of the important results that Payne measured was that the laser was constructed using a 2 micron diameter fiber. So by our measure, we should be very close to that number, maybe a micron bigger, but in theory, we should be able to still create a laser with that fiber. And they achieved a 28 milliwatt lasing threshold. So that was the minimum power needed to actually create a laser. One of the issues that we found so far is that our diode laser, it's about 60 milliwatts. And then through coupling into the fiber and also the fact that the diode laser excites a lot of higher order modes within that actual laser, we're actually not seeing anywhere close to 28 milliwatts being launched into the fiber. So we're using a new laser now that should allow us to get up to this 28 milliwatt level and in theory create a laser. So dye laser physics really quick. So the dye medium we're using is an organic liquid dye, rhodamine 6G, and that is the gain medium. The interesting thing about these liquid dyes and organic liquid dyes is they can be used to laze at a variety of wavelengths. So for instance, we're pumping our gain medium at 520 nanometers, but the fluorescence region of rhodamine 6G extends from about 570 nanometers to about 590 nanometers. And the structure of the atom notionally is that we excite at about this level. So this is in theory a 520 nanometer transition. And then this transition down here, after we go through some vibrational transitions, is in between that 570 and 620 nanometer range. These dyes normally require continuous circulation to laze effectively. That's when they're being used in higher power setups. But in our setup, we believe because we're using such a low power and we're interacting with so little of the dye, we don't think we'll actually need to circulate that dye through the cavity to create a laser. So our plan for the optical cavity, we start out with the 520 nanometer diode laser that's launched into the fiber. It then propagates through the fiber until it reaches the taper region, which is submerged in the gain medium. This is where that evanescent wave interacts with the gain medium, and some of that fluorescence from the rhodamine 6G couples back into the fiber. So once it's in the fiber, it is then reflected by a dielectric mirror butted up against one end of the fiber. It then propagates back through the gain medium. That fluorescence then stimulates emission. And then we have this 567 nanometer short pass dichroic mirror. So that allows the 520 nanometer light to pass back through the mirror, basically exiting the cavity, while the fluorescence is reflected off of that mirror into an output coupler with the reflectance of about 0.95. So then this output coupler and the fiber and dichroic mirror all the way to the dielectric mirror make up that laser cavity, and the laser output comes out of the output coupler. In this setup, we actually won't be able to select for a smaller line width or a specific wavelength to laze at. The laser will basically be lasing at a wide line width somewhere in that fluorescence range, but that won't be able to be selected by us at this point in time. So for fluorescence results, so fluorescence has been coupled back into the fiber. You can see that here, this is measured by a red tide spectrometer. So after being passed through a few dichroic arrays to try to filter out as much of that green light or that 520 nanometer light as possible, this is the spectrum that we see. So you can see the 520 nanometer peak here. And then between about 590 and about 620, we see the fluorescence spectrum of the rhodamine 6G. With the dielectric mirror butted up against one end, we are reflecting a noticeable amount of the fluorescence, but that is one of the problems we're addressing right now is that back reflection of the light through the optical fiber. And in order for more fluorescence coupling to occur, we need a thinner fiber, which again, we've addressed a little bit recently. And we also need more pump power, which I've also talked about. So for some of the experimental challenges that we faced so far, so some of the things we're really trying to fix with the setup and eventually create a working laser. So we noticed our dye concentration was about 20 times too high as compared to the Payne paper where they created a working dye laser. So our concentration was about 2 millimoles versus the Payne paper, which cited 0.1 millimole concentration of the dye. So that's something we've addressed. And we also noticed that with these smaller fibers, as soon as we put them into the dye solution, all the light coupled out. And this was because the ethylene glycol had an index of refraction of 1.45, while the fiber cladding had an index of refraction of 1.46. And because these numbers were so close, the total internal reflection that is used to actually keep the light inside the fiber was not as effective as we needed it to be. So we're using ethanol now to dissolve the rhodamine 6G, and that has an index of refraction of 1.36, which should be much more effective. For the back reflection through the fiber, we did see an appreciable amount of back reflection, but it's really not enough to actually create a laser. So we're using this new back reflection technique where we have instead of just the dielectric mirror being butted up against the end of the fiber, we use a collimating lens and then reflect that back into the fiber. So in theory, that should give us a few more kind of degrees of freedom to actually get that light back into the fiber. And then also we're using a lock-in amplifier just because the power of this light is so much lower than the pump light that's initially being launched into the fiber that we really need a way to see these very low power changes in the fiber. And so we're using that lock-in amplifier to see that try to measure some back reflection. So just a quick summary and where we're going in the future. We've achieved these single micron diameter optical nanofibers routinely. Again, you see we're about at the 3 to 4 micron range, maybe a little smaller. We really can't tell with our current setup. We've used these tapered ONFs to excite the rhodamine 6G laser dye, and this was observed in the fiber output. So in the future, we're going to try to maximize this fiber fluorescence output with these smaller tapers, new laser, a few of the other things that I talked about that we're addressing. We're going to construct this cavity around the fiber. This is basically done, but we need to address the back reflection issue before that's finally complete. And in theory, at the end of this project, we'll be able to achieve a measurable lasing gain. Does anybody have any questions?
Awesome job, Zach.
Thank you.
That's fun. That's cool. I really like the picture of the glowing yellow laser thing. That was awesome. Okay, so looks like Jacob.
Hey, good presentation, Zach. I was just curious, how are you actually physically mounting your fiber inside of your die? Are you just submersing it in the solution, or?
Basically, yeah. So we have this little trough built here, and we actually have a new trough that's bigger and deeper, and just will allow us to have more dye in the trough. But we essentially just tack the optical nanofiber at both ends. You can't see the other end here, but it's just basically glued in there. And then yeah, so the dye solution is just filling the trough, and we just basically lay that fiber into there, and then the evanescent wave just interacts with the fiber from there, or the dye from there.
That makes sense. Thanks.
Yep.
Thanks, Jake. Who else has a question? Okay, well, so Zach, how far does the evanescent wave extend outside the fiber?
So that extends about 250 nanometers outside of the fiber. And so you can see that when basically the laser is within the core, that extension comes out into the cladding just a little bit, and you're essentially seeing nothing outside of the fiber. But once we get into the taper region where the diameter is only about 1 micron, that 250 nanometers that the laser actually extends through that evanescent wave into the gain medium is an appreciable amount of the laser's power. And that's why having these thinner fibers is much more beneficial to actually exciting that gain medium, just because more of that laser power actually exists outside of the fiber.
Neat stuff. Who else has questions? I just have so many I want to ask. No. You guys gonna get like a new microscope so you can I don't know, it sounds like you have limitations on that.
Right. So there are, in theory, a few adjustments we could make. That's just not something we've been focused on right now because we're addressing a lot of the other problems that are in our setup. But as soon as we get back to the point where we're pulling these nanofibers again, basically once we solve the back reflection issue, then yeah, that will become a main focus because we really want to see actually how big these fibers are. And so there are a few adjustments we can make to the camera and microscope setup.
Cool. Anybody else? Oh, we got a couple more minutes. You guys are like my 215 class. Turn on your videos. Actually, speaking of that, I'm really glad you talked about all the Snell's law kind of stuff. Now I'm gonna show my class look, this total internal reflection is important. So I really like the practical example there.
Right.
So how are you changing that algorithm to optimally get the thinnest cable you can get?
Yeah. So again, the code that actually makes that algorithm was written by Hoffman in 2014. But we essentially define a few parameters. We define parameters based on our actual experimental setup. So what we've actually made in the lab. So a few of those parameters, like the flame width. So obviously the flame isn't just a single point in space, and that actually influences how quickly the fiber is stretched across that flame. And then also one of the things we have to be careful about is how close to the fiber that flame nozzle actually is. So if you think the flame is not this perfect cylinder or perfect rectangle in space, it changes diameter as you move closer or away from the flame nozzle. So that's just something we have to be careful of. And then in that program, we actually define sort of our final desired parameters. So what angle the taper basically starts at. So I'll go back. So like what angle this comes down at, how long this region is, what the final actual diameter is. And those inputs that we give to the program are kind of notional. And we're trying hard to actually match those up to what we get in real life. So the program we're using to pull these fibers right now, in theory, it gives us a 500 nanometer diameter fiber, but actually it gives us maybe a 3 or 4 micron fiber. So we're really trying to match up what we're giving to the program and then what we're getting out in terms of a real life fiber.
Cool, really neat. Any questions? Well, thanks, Zach. Great talk. Woo.
Thank you.
Okay, next up, we have C3C Madison Marsh. We'll be presenting on a limiting magnitude study of the Virgin Islands telescope. Sounds like a nice vacation spot, maybe, Virgin Islands. Okay, I see your slides, and I hear you.
You can hear me?
Yes.
Okay, I didn't know if my
Perfect.
mic unmuted. Okay. So, like you said, good afternoon, everyone. I am C3C Madison Marsh, and I'm representing the United States Air Force Academy. And today, like he said, I'm talking to you about a limiting magnitude study of the Virgin Islands Robotic Telescope. So underneath my name, I have the other contributors to this study. So first off, Dr. Giblin, who is my research advisor, and then some of the other ones, Priya, Bryce Orange, and then Dr. Devin Delarose, who's also at the Air Force Academy. So for the overview, I'm going to go over the project objectives and then talk about the probing of gamma ray bursts and gravitational waves and potential progenitors. And then I'm going to talk to you about the actual characteristics of the Virgin Islands Robotic Telescope that we did this study on. Then I'll talk about the actual standard Landolt field we used, some data analysis, results, and then of course the conclusion. So for project objectives, we performed a limiting magnitude study, and it was using a standard Landolt field that I picked out, which is Rubin 149, and I'll get into that a little bit later. And then this reveals how deep we can image in our field of view for the Virgin Islands Robotic Telescope, which is actually trying to say how faint we can actually see. And that kind of leads into what we can actually follow up in for gamma ray bursts and gravitational waves. If we know how faint it is, then we can decide which signatures we're going to follow up for optical imaging. So for the production of GRBs, so this is a artist rendition of a binary merger. So that's either a black hole, neutron star, or neutron star, neutron star. So within these, for gamma ray burst, this is depicting a release of gamma ray burst and gravitational waves. So gravitational waves, there's 2 types. We have short, which is less than 2 seconds, and then long, which is greater than 2 seconds. So for these extremely high energy events, it's postulated that these really large mergers are what is causing these gamma ray bursts and gravitational waves. So for the Virgin Islands Robotic Telescope, it's 0.5 meter in diameter. It's a F10 classical Cassegrain. It's on the island of St. Thomas, and it's actually geographically located in a strategic position. So it's the farthest east in North America, and it's closest to the equator. It's got a CCD camera on it, and you can read some of the characteristics of it as well. And the filters was Johnson Cousins, and we did our imaging in the R band. And you guys can take a look at these nice photos we have of the telescope that we used. Going into the standard Landolt field, I did Rubin 149, and we took the images in using the R filter. And whenever we chose these, it was a standard star analysis done by Landolt in a really famous 1992 paper that he released. So when going about this, there was 30 images that were taken at 10 seconds each, and it was an effective exposure time of about 300 seconds. And then properties, once again, this was taken from his 1992 paper. So you can see these are all the list of all the properties of the stars. And if you look on the right side of the page, there's the picture. And then that little green circle at the top shows the stars that we actually pictured in this Rubin 149 field. Getting into source extraction. So in order for us to actually calculate the limiting magnitude, we need to determine how many sources are in the image. So what we're going to do for the stellar profile under source extraction, we're going to obtain a full width half max using a point spread function, which is a radial plot routine using a Gaussian function around 10 sources over the range of brightness. And then we're going to exclude any saturated sources. Then we determine the mean of the full width half max and of the point spread function. So then we go into source extraction for the DAO find algorithm. So we're running that DAO find routine, locating a density maxima, and then we're specifying a signal to noise ratio, which was 5 in this case. And then we're outputting a coordinate file. So within this, we detected around 1,200 sources, which is a lot. And then going into aperture photometry, this is just measuring all of the brightness of the sources that we photographed. So for aperture photometry, we're setting an aperture size about 2 times the full width half max. And we're going to set an analytes width, and that depends on how crowded our field is. In this case, we had a really, really crowded field. And then our input will input a coordinate list from the DAO find algorithm, output an ASCII file, and then we're going to dump all of the and extract coordinate positions and instrumental magnitudes, and we'll read it into Excel. So then after we read all of this information into Excel, we do some magnitude calibration. So we identify the standard Landolt field stars that are in the field that we photographed. And then I locate these standards in my spreadsheet, and then I'm going to use the instrumental magnitudes and the published magnitudes to determine my ZMAG. And then I add that ZMAG to all of my instrumental magnitudes in my spreadsheet, and that's how we get it calibrated. So this is the first slide of our results. So this is R magnitude against R magnitude uncertainty. And you can see there's a horizontal asymptotic relation, which would mean that the limiting magnitude, as you can see, it approaches around 20. So that's what we would say for the Virgin Islands Robotic Telescope, that it's estimated to be around 20. For this next one, this is a histogram against the number of sources detected at a certain magnitude. So let's take the R magnitude 15, for example. You can see it has that number 63 above it. So that means that there are 63 sources detected to be at the 15th magnitude. So for this photo, as you can see in the histogram, it starts to kind of drop off around 18, but it does go until around 21. We think the reason that this isn't in complete correlation to the graph prior is because we photographed such a field that was really overcrowded. So maybe some of our sources that were actually supposed to be detected got lost in the background noise. So overall, for the conclusion and future work, like I said, the limiting magnitude in the R band is 20 for the Virgin Islands Robotic Telescope. And what we want to do next time is we want to carry out this project on a less crowded field, because we saw obviously in the histogram that overcrowded fields can really mess up some of our data. And so we want both of them to be in congruence with one another once we do it again. And then now that we know the limiting magnitude of the telescope, we want to do follow-up observations for the optical afterglow of gamma ray bursts and gravitational waves within our limiting magnitude. And then additionally, we want to adopt this study for the USAFA 1 meter telescope in the fall of 2021. So now I will open it up for any questions for anyone that has some.
Great job,
Hey, Madison.
Thank you.
Madison, this is Dr. Chun. Very nice presentation you had there.
Thank you.
I have one question and one request. The question is, are you going to do this limiting magnitude study also for the other 3 Johnson-Cousin bands?
I believe we could, but in this case, we only did it in the R band.
Okay. And then the request is, in addition to the USAFA 1 meter telescope, can you also do this, please, 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 to the Falcon Telescope Network?
I would honestly love to take on that project if that's something that I can get my hands on. So.
Oh, absolutely. And you don't actually have, the Falcons are automated as well, so you don't have to operate the telescope.
Yes.
Okay, Dr. Giblin said definitely, so thank you.
That's great. Who else has a question for Madison? Okay, Madison, could you talk a little bit more about how the images were processed?
Yes, so I'm going to go back to the slide that shows, okay, so this was the image that we used for this presentation. So basically, once we take these pictures, like I said, the 30 images, we're going to take that raw image and it's going to be processed in just Python. And then once we take those processed images, we're going to do some dark image subtraction and then flat fielding. And then we just stack all of them on top of one another. And then we get this beautiful photograph that you guys see here.
So how long does the whole process take? Is it mostly automated or?
I didn't do the processing myself. That was done by Priya, who works at the telescope. And I don't believe it is too entirely time consuming from what I understand, but the beginning of it, yes, is all automated.
Thanks.
Nice job, Maddie. What's the, you might have said it in the beginning and I missed it. What's the size of the telescope?
Yes, so it's a 0.5 meter telescope.
Oh, okay. Out of curiosity, so if you do this on the 1 meter, do you know then about what magnitude to expect for the USAFA one, or?
I don't know about the magnitude for the USAFA one, and I would assume maybe the location of it might also play into how far it can see. So like I said, since this one's strategically located, that also might have a factor into what magnitude. So I really don't think I would know too much about what to expect on the 1 meter as of right now.
All right, thanks.
Hey, Madison, this is Dr. Chen again. Just another comment. I don't know if you got to see C1C Ethan Albrecht's talk earlier this morning, but we do have some BVR data on some of the falcons that he has collected, trying to come up with the extinction curves. But so some of that data, you can use now to help us determine the limiting magnitude for those telescopes.
Oh, okay, awesome. I think I did catch most of his presentation this morning, but I was having a lot of internet difficulties, so.
Great, thank you.
Thank you.
Yes, gotta recruit you over the Caesar research. So could you talk more a little bit more about how the location, you said it's strategically located, like what kind of factors go into that?
So the factors that I mentioned was it's the farthest east in North America and closest to the equator. And I believe that what that does is we can see more, I guess, into our atmosphere and at the poles. So I guess we can have more of a wider range of view for what we can photograph and image in the sky.
Does your group have any research funds to send you out to the Virgin Islands to?
Well, actually, I was supposed to go out there for 2 weeks at the end of the summer, and some of the summer scheduling here kind of messed with those plans, but we're still trying to get me hopefully back on track to potentially get a TDY to at least go out there for a week to do some follow-up studies with them.
Oh, wait a minute. If it's automated, you don't have to be there.
Yes, you do, sir. Just like you don't have to go to Hawaii for the AMOS conference. It's all virtual. Let's see. You also talked about for future work, picking out a less crowded field. Like what kind of factors would go into that? Or like how would you go about determining what that is?
So I believe the reason why this one was more crowded, it was I got to choose the fields that we were going to photograph to take the limiting magnitude from. So I guess my suggestion would be, before I guess we get into the process of actually taking the images, maybe picking out several different fields first and doing like prior follow-up studies to see what those fields look like before we start the limiting magnitude process. Because there's, oh, sorry.
Go ahead.
I was going to say that there's the list from the Landolt paper, the 1992 paper, the really famous one. There are so many fields to choose from. So I don't think it would be a big problem. What I did whenever I chose the 2 fields, which we only included 1 for this presentation, just for time's sake, what you would do is you look at the the correlation in the sky to your location and where you can see. So that's another factor that would also play into choosing one for the next time around.
Thanks. And could you remind me again, why are you choosing it from the 1992 paper, like that same region? Is it just like really well characterized and understood, or?
So this Landolt paper is used basically for any sort of standard star analysis. He took almost all of the data for standard Landolt stars, and that's why they're called Landolt fields from what we chose our study from. So he has, like I showed here in this properties page, he had all of the properties like all of the different bands of filters, or the bands that which you can image, and then declination, anything like that. So it shows all the different ways that you can view these fields, I guess. So he has a bunch of information that we can take from to do more studies on.
Great, thanks. Does anyone else have any other questions? I'm excited for you. You have another 2 years ahead of you to do all this research. What do you want to do?
Yes, me as well.
Do you have an idea what you want to do after the Air Force or Space Force? Or I mean, as after the academy?
Yes, so once I leave here, I'm hoping to get one of the graduate scholarships to go right in and get my PhD in high energy astrophysics. And then once I get finished with that, I want to become a fighter pilot and then hopefully become an astronaut is the end goal. And then
just want to do everything.
Physics research.
Yes, great goals, great goals.
Thank you.
Let's see. Okay, well, I guess we can transition a little early because our next speaker is ready. We'll just slowly transition, unless anyone else has any other comments or questions. Okay. Well, good job again.
Thank you.
Madison, great job. Excited for next CSURF next year.
Thank you very much.
Have a good one. So Raymond, are you just setting up? You feel free to go ahead and share your screen. We have a few minutes though.
Yes, sir, already.
So Raymond, I want to give a bit of more of a background about yourself. So this is C2C Raymond Baum. This is on event-based sensing of satellites, but just what you plan to do in the Air Force and how you've enjoyed this research.
All right. Well, like Major O'Keefe said, I'm C2C Raymond Baum. I'm in Cadet Squadron 2 here at the United States Air Force Academy. Yeah, just a little background on myself. I was in Squadron 18, now I'm in Squadron 2. I'm a physics major. I'd love to go pilot after this. I want to go fly F-15 Eagles and see where life takes me from there. But yeah, this event-based sensing is what I'm going to talk about today. And it's really been the thing I've been doing since sophomore year. I started working with Major McReynolds when he first got here. And then I'm currently working with Major O'Keefe in the physics department for characterization of the Davis 240C and the Prophecy third generation cameras. So just getting into it, I'm going to just start with kind of the motivation behind why we're doing this. Space has become increasingly complex over the past 20 years. So you think about, in the beginning, it was really just military use, GPS satellites, and things like that. But now with kind of commercial satellites being available, we've got CubeSats, we've also got the FalconSat from the Academy, we've got commercial like radios, and you think of like Dish TV and satellite radio and things like that. So that just adds the complexity of low Earth orbit. And I'm sure a lot of you know the Kessler syndrome, which is one collision causes a chain reaction that just completely obliterates everything and renders low Earth orbit completely useless to us. So our motivation is, how do we track all of this effectively? Like, you think about the current methods to track things is very, very expensive. Like, you go out to Schriever and Buckley Air Force bases, those big domes are the current satellite tracking apparatus, and they're big, they're expensive. And so we just want to look for a cheaper way to do it, a better way to do it. And that's where we think event-based sensors can come in. this is the overview, kind of what I'm going to talk to you about. I'm going to first introduce what exactly event-based sensing is. I'm going to talk you through our testing setup and methodology, then work through results, conclusions, and then our future work, what we want to do from here. So, an introduction to event-based sensing. What is it exactly? So pixels operate independently and activate only when there is a set % change in the threshold, which is not a set value. It operates on a log scale, which is a % of the current value. And so what we find with event-based sensors is that data is only recorded for active pixels. You only pick up things that are changing, and you only pick up data that's changing and pixels that are changing. So essentially what we're doing is we're not collecting redundant data from unchanging backgrounds. So right here is kind of the output that every event-based sensor will give you. It's an X, Y, timestamp, and a polarity stamp. The X and Y are just basic coordinates, depending on the camera's field of view. The one I'm going to talk about is 240 by 180 pixels, and so these are just coordinates for which pixel fired at which time. This is in seconds, and polarity for 1 means an increase in luminosity, and a polarity for 0 means a decrease. So it's just a binary on or off. The advantages of event-based sensing is shorter latency period, a much faster stimulus detection, about 1/100th of a traditional camera, much lower power requirements, and a much wider dynamic range. So what I'm going to do here is play this video, and what you're going to see is just the event-based output from a drone, from our friends at Western Sydney University. They recorded this some time ago, and they sent it over, and I just think it's interesting, and it really highlights some of the things that an event-based camera has over a traditional camera. So one thing you're going to see is the sun coming to the field of view of the camera. And with the traditional camera, it would go out of focus with that. That's due to the dynamic range. But because the event-based sensor has a very, very high dynamic range, you'll be able to see the sun, and then you'll be able to see everything else. So you see the two people waving right there, and then you see the sun. You see some mountains in the background, and then that's really output. You saw, I mean, kind of in the snapshot right here, you can see the outline of the mountains, but where the mountains are, there's not really any data coming in. That's because it's relatively unchanging compared to the surroundings. So our testing setup and methodology, essentially what we did was we took an independent Davis 240C and co-sighted it with a USAFA telescope. It had a 25mm lens on it and 18.5mm pixel pitch. Like I said earlier, it's a 180 by 240 pixels with a field of view of 7.6 by 10.2 degrees. And then each individual pixel has a field of view of 0.042 degrees in both the horizontal and vertical directions. So essentially what we did was we obtained satellite pass information from Celestrak. We got the TLE data, and then we used the Sky 6 software to point the telescope at the region of the sky where we knew that the satellite was going to pass through. And then what we did was we just held the telescope apparatus there and just let the satellite pass through. And then we were able to gather some data, and that's our testing setup and our methodology. And like before, we used the JAIR software. It just comes with the Davis 240C. It's in Java, and it just helps streamline the entire process. So the next slide I'm gonna just play a video is the actual output of the Davis camera, and you're gonna see a few things. You're gonna see about 3 satellites pass through the field of view. This is a filtered image, so it's not exactly what the camera saw and exactly what the JAIR software saw, but it is quite a good approximation. All right, so you saw the 3 satellites pass through, and that was essentially what we saw on the JAIR when we had our testing setup, and then we kind of moved everything over to MATLAB, where we got more analysis done. So the results. This is a full-time integration. So you can see right here we've got 2 satellites that pass through the field of view, and I think really interesting thing with this is that just how much more noisy the positive events were. Positive as in means on again, and negative means off. I would use those interchangeably. But another thing is a pattern noise in the positive events. That was really interesting to see, and not so much in the negative events either. Additionally, you can see that 195,000 positive events were detected compared to 30,000 negative events detected. That's a crazy difference in just how many events were detected. Note that we do not see all 3 satellites. You may have seen a 3rd satellite in the previous slide, but we did not see that. That kind of indicates to us that it was not bright enough to detect with our relative bias settings or things like that. Right here is a histogram. That's basically the time integration from last time, just flipped over into a 3D plot. And you can really see that certain pixels are noisier than others, especially when looked at from a positive and negative standpoint. And this really shows the location of those pixels as well as their relative noisiness. Note that we do not see any traces through the histogram of the positive events. If you look at the negative events right here, we see the X marks, I guess X marks the spot by the 2 satellites tracked, but we do not see that in the positive events. That kind of indicates to us that the negative events are less noisy, which is something that we also gathered from last slide as well as this slide. All right, kind of moving on to an event rate versus time plot. That's something that we thought was really interesting and something that we thought would be very, very helpful, and it ended up being helpful. So essentially what we did is we took a no signal region and a signal region, took it for multiple time integrations. So we did 3, 1, and 250, 3 seconds, 1 second, and 250 milliseconds. And essentially what we did was just integrated the events per time, and we would get our event rate versus time plot. This is kind of just to show you visually what that looked like. These are the results of the event rate versus time, and you can kind of see different integration times give different resolutions as to when the event happens. So you look at the 3 second, it's kind of wide, the 1 second is narrower, and then the 250 millisecond is actually fairly good. And same with the negative event integration times, 3, 1, and 250 milliseconds. So the event rate was done on a 20 by 20 region of pixels where the satellite was known to have passed through, and then another separate region where the satellite was not known to have passed through. And you can kind of see just from the other slides as well, that kind of carries over, there's much less noise in the negative events than there is in the positive events. So kind of taking that and turning it into a signal to noise ratio, we have 2 signal to noise ratio plots here, one for the positive events versus integration time, one for the negative events versus integration time. So the kind of the takeaway here is that the SNR increases as integration time decreases, and the negative events have a signal noise ratio of around an order of magnitude higher. So really something that we kind of need to look at is, why is the negative events SNR higher than the positive events? We think it's a function of the bias settings, which I'll kind of talk about later, but we just need to figure out why the SNR is higher on the negative than the positive events. So in conclusion, we had a successful detection of a magnitude 5 satellite. And for those of you that don't know kind of relative magnitudes and things like that, a lower magnitude represents a brighter object. For example, the sun has a magnitude of -26.7, and that's the brightest object in the sky. So that shows us that, we can get a pretty good satellite, a pretty important satellite with quite low and things like that. And then the positive total integration time shows vertical noise bands. We're kind of interested in that as to why that occurs, if that's just a function of how we're analyzing it, or if that's just part of the camera and how that works. Noisy pixels are evident in the histogram, and however, this is an inconsistency between positive and negative events. If we go back to that histogram, the same pixels will not be hyperactive on the positive or negative side. So we need to figure out why that is. And additionally, as I said, the signal noise ratio appears to be better for negative events. Again, could be a function of the bias settings. So future work, things we'd like to do is optimize bias settings to boost the signal noise ratio, particularly in the positive events. Use a new lens, 85 millimeters, zoom in a little more and get a better area, as well as the next generation camera, which is the Prophecy. Something else we'd like to do is kind of incorporate more or better data processing algorithms, which is software noise filtering, automate satellite detections, and then with line detection algorithms, and then possibly bring in machine learning to detect these things. And then essentially what we were doing was prepare the Davis to look for the ISS to look for upper atmospheric sprites in our kind of broader view of hypersonics detection and missile defense, things like that. Any questions?
Yeah, Raymond, this is Dr. Chung. I wanted to jump in before I know Major O'Keefe would ask a question. So I actually had two questions. But before that, I really enjoyed this talk because I really wanted to see results from what you guys were doing in the observatory. So I did have a question about those two histograms, positive events, negative events. And you answered one of the questions as to the pixels didn't quite match up. So where I see a lot of positive events at a given pixel, I don't necessarily see a lot of negative events at that same pixel. But what percentage of the pixels in your focal plane actually had both positive and negative events? Do you know?
I do not know about that, sir. I think that's a really good point you bring up because, I mean, if the positive events are going off, I guess those same pixels won't have enough time to register a negative event or things like that. But I think that's a really good point that you bring up.
Okay, and then the second thing is, even though we can see the satellite crossings in this histogram of the negative events, why is it that we can pick it up on those colored images on the positive events, but not on the negative events? And I think you mentioned something about could be a bias setting.
Yes, sir. So the bias settings that we use essentially change how the camera responds to certain inputs and how the pixels respond to certain inputs. And the bias setting that we use was more for low light settings and high frequency settings. So it could be a function of that. We're not really sure, but we would like to find that out.
Great. Thank you very much, Raymond.
Thank you, Dr. Chun. Does anyone else have some questions for Raymond? In the meantime, what are those? Why are there red spikes there in this picture?
So those red spikes are indicative of how much noise there is. So kind of look right here, kind of goes over to about 1800, 1600, 1600, 1800. So that's the events. That's how many events occurred. And you can see for some of these positive events, there's a lot of events happening there, which really means that it's just a noisy pixel. That pixel just picked up a lot of noise there.
So a single pixel, a noisy pixel?
Yes, sir.
Okay, could you go on to the next slide? I think that. So you mentioned a little bit about pattern noise in the image, and I guess I'm trying to understand, I guess, the reasoning or what, as far as like analyzing the signal to noise ratio by picking the middle of, part of that little square. I don't know if this is just, illustration purposes, half of it's in band, half of it's in the pattern noise, but Can you speak to that maybe a little?
Yeah, that was really just for illustration purposes. It's the same size box, just in different areas of the field of view. But I think something that we also need to work on for future work is using the no signal region and the signal region as the same region. And so kind of bringing up the noise that's there before the satellite passes through, and then the signal when the satellite passes through. I think that would give us a cleaner event rate versus time, and then a cleaner signal noise ratio, whereas looking at two different areas may be different.
And do you expect that might change if you're like, oh, I'm right in the middle of this pattern noise?
Yeah.
Who else has some questions? Okay, well, just go through my list. We'll see. So you talked a little bit about how your magnitude 5 satellite or seeing one of those, how significant is that? What can I see visually with my own eyes? How does that compare?
So like I said, the sun has a magnitude of negative 26.7. However, the naked eye limit is about +6. And that's in completely dark areas with no light pollution, nothing like that. And that's just kind of an average of humans. Some people see better, some people see worse. But so this is in the naked eye, but for some people.
So you're telling me my eyes can see about as good as this camera, this fancy dancy camera. Oh, so what are some practical reasons for applications? Because you're actually going to be briefing at the CERB here in a few weeks.
Yes,
high level, at the DF level. So what are applications, or why is the Air Force, the DoD, interested in?
Yeah, so we're looking at using this for hypersonics detection, and so just finding really fast events. So a traditional camera would need 10,000, 11,000 frames per second. That requires a massive amount of data just to bring in. Whereas one of these cameras, you don't need quite that. You need a lot, but it's nowhere near what a traditional camera would need. So finding very fast events, and that's kind of where the next slide goes in, is we're going to be looking for upper atmospheric sprites, which are very, very dynamic events. They just happen on and off. And so a neuromorphic camera, we think, would be ideally suited for detecting hypersonics.
So where does this data savings come from?
Well, it comes from the fact that, like I said earlier, the redundant data, or the areas in the field of view that aren't changing, do not get picked up by the camera. Whereas in a traditional camera, you take everything in the field of view, and even the things that aren't changing, things that you aren't really interested in, get picked up. Whereas for this one, things that are picked up are changing.
Yeah, one more question, Raymond. This is Dr. Chan. So do you know what the second satellite is that you picked up and detected?
I believe we do, but I do not have that information with me right now, sir.
Okay. And I assume if you go back to that slide that shows the table from some website.
Yes, sir.
That one. So the 5 magnitude is what they estimate the brightness of the satellite would be at the maximum altitude. Is that where you try to capture it, or did you capture it some other part of this orbit, of this pass over the telescope?
I believe we captured it at a different point. I don't think we were necessarily looking at the max altitude.
Okay, because that would really be important. How dim can you see?
Mm-hmm.
You weren't really looking for a 5 magnitude source. You really wanted to see, if you could have seen it at 6 magnitude or even 7 magnitude.
Yes, sir.
Okay, could you go back to one of your later slides? I guess it doesn't really matter where. So you talked about the next generation camera. What's different between the Prophecy, which is a generation 3, versus your Davis? And
yeah, so The Prophecy is 480 by 640 pixels, so it's a much larger field of view, or I will say it's a larger focal plane, and so we can see kind of better things with it, and then kind of the architecture is better, the programmatics, the software, the data collection software is better, and so yeah.
But is the software better? Just teasing, because yeah, we're having troubles with the software right now, right?
Yeah.
So what else is good about the Prophecy compared to the Davis in terms of like your positive events? It was a lot more difficult to, I don't know, to determine things with all the. I don't want to give the answer. What else is?
Yeah, so I mean the noise we think is better on the Prophecy. So it's just kind of an improvement on the Davis, and we think a lot of the issues that we saw, particularly with the noise, will be reduced with the Prophecy.
Oh, could you briefly talk about what you plan to do this summer?
Yeah, so this summer I'm going to go down to Albuquerque and work with AFRL on Prophecy data collection with Dr. Dave Monet and Dr. Peter Crabtree. And we're going to kind of do a continuation of this, and then but kind of use the Prophecy as well. So that'll be a really interesting project. I'm really looking forward to that. So,
great job, Raymond.
Thank you. Appreciate it.
Enjoy the rest of your Saturday. So next up, we have C1C Audra Jensen on characterizing satellites using near simultaneous polarization measurements.
Yeah.
Ooh, cool space background. I like it. I grew up in space. Oh, and your Space Force tags. I like that.
Yeah, I got my blue tags.
Congratulations.
Thanks.
Yeah, feel free to share your slides. I see the space. Oh, there we go. See the slides? You're good to go.
Awesome. Okay. Hello. Good afternoon, I guess. I am Cadet First Class Audra Jensen from the United States Air Force Academy. And today I'll be talking about the research I'm currently doing, which is characterizing satellites using near simultaneous polarization measurements. Cool. Here's a list of my mentors. Big thanks to them for helping me out the last year on this project. I've had a lot of fun doing it. And then, so we have the overview. So for this presentation, I'm going to start out with the motivation behind this project. Then I'm going to talk about the objectives and tasks for the project, the theory behind it, the methods we use, what people have done in the past, and the work I did this semester, and what I'm going to try to finish up this semester, because this is my last semester at the Air Force Academy. Okay, my objective is to characterize geosynchronous satellites based on near simultaneous polarization optical measurements. My tasks were to reduce and process many satellites' polarization signatures, which was done using a program called MirrorPro and pre-existing MATLAB code. I also wrote a lot of my own code to continue with analyzing the data. I have been able to observe more nights of data this semester so far, and I plan to observe more, maybe more, before the year is over. In the future, we'll be using my findings in order to compare and analyze my data with respect to satellite attributes and characteristics. Cool. So I'm going to start by talking about some of the theory behind my project. So the first is base object characterization. Result imagery can provide us information about a satellite's characteristics, but at geosynchronous orbit, very large apertures are required to resolve objects. This is actually an artist rendition of the geosynchronous communication satellite WildBlue 1, which is one of the satellites we've observed a few times. The picture on the right is what WildBlue 1 actually looks like in the images that we took using our 16-inch telescope that we have here at USAFA. So this is not a resolved image. It just looks like a point of light. You can't even tell that it's a satellite. We have to treat satellites as unresolved point sources, and to characterize satellites with this information, we have astronomical techniques we can use. So for my project, we use polar imagery on the images taken with our telescope. So here's some more of the theory behind our observations. We have Brewster's angle, which is the perfect scenario when unpolarized light glints off a surface and the reflected ray is completely polarized. This angle is different for different surfaces because of different indices of refraction. We also have Malice's law, which is unpolarized light goes through a generator that makes the light polarized. Then this polarized light goes through an analyzer, which filters the polarized light to a certain angle. So we're able to record the information about that light coming out of the analyzer, and we have to do some calculations to work backwards and find out what the polarization of the original generated light was. This is the form of a W matrix, which is our calibration matrix for our telescope. And then we also have Stokes parameters, which is how we describe the linear polarization of light. And it can be fully characterized by 3 Stokes parameters. So the S0, which I'll be talking about a lot, is going to be the total intensity of light. The S1 is the preference toward the horizontal and vertical polarization. And the S2 is preference toward the diagonal directions, so the 45 and 135-degree directions. Okay, so this is our experimental setup. We use our EUSAFA 16-inch telescope to view clusters of communication satellites in geosynchronous orbit. The light we are observing has been changed several times throughout the process. So the light comes from the sun and is partially polarized by the satellite. We receive light in our telescope where it bounces off a primary and secondary mirror, and then it goes through our chosen polarization filter and finally hits the CCD where we record it. We use 4 polarization filters, so we have a 0-degree filter, a 90-degree filter, a 135-degree filter, and a 45-degree filter. After an image was taken, we would switch the filter so there's about 4 to 8 seconds in between images with different filters. We record the intensity out at the CCD and use that calibration matrix to convert it to the input Stokes parameters. So here are the polarization observations that were taken in the fall of 2019 and the spring of 2020. We're still processing and finishing off the data from the fall of 2020 and now the spring of 2021. There are 10 nights of observations observing 14 different satellites. The green indicates a strong glint if it says glint, but if it just says glint and it's in black, it means it's a weaker glint. And the red indicates data that we removed from our analysis data set because of atmospheric turbulence or another atmospheric effect that have corrupted our data. And so that's going to be discussed more in detail later in my presentation. So before I get too deep into my data, I first wanted to show this time series plot. I use a lot of plots like this in my presentation, but they'll be much smaller, so I wanted to explain the axes and what I mean. So first, there are 3 colored lines in this graph. The black is the S0, so the total intensity of the light. This is in raw counts per second, which you can see on the right side of the axis. Then we have the S1, and so S1 is green and the S2 is red, and both of these are normalized to S0 as seen on the left y-axis, which why both of them stay near 0. The x-axis is just time in UTC. If you see a gray vertical line on the graph, which isn't in all the graphs, it just indicates where a glint starts. As of right now, the glint is defined as 15% of the maximum intensity. For the most part, a glinting satellite will have a raw normalized counts of around 10 to the 6th. A weaker glinting satellite will be around 10 to the 5th, and a satellite that did not glint will be around 10 to the 4th. This is still subject to change, but this is currently my criteria. In our analysis, we focused first on the satellites that glinted in the spring of 2020. We were comparing them when we noticed that the data for Spaceway 3, which is the one on the bottom right, was different from the other satellites. There was much more variation in both the S1 and S2 that was present than any of the other satellites. To check this, we plotted the S1 and S2 for all 4 of these satellites on the same graph. As you can see, other than Spaceway 3, all the other glinting satellites were not polarized more than about 0.2 in any direction. This confirmed that Spaceway 3 appeared to be much more polarized than any other glinting satellite, which was a little bit suspicious to us. This data was collected on March 12, 2020. Our observation notes indicated that there were some high clouds present that night, and this could mean that the data could potentially be corrupted. The next question we wanted to answer was, if there are clouds in a data set, would all the satellites in the image exhibit the same dynamic features? So to test this, we decided to compare not just the individual satellites, but the clusters. These 2 clusters were observed on 2 different nights. The one on the left is from the 3rd of March 2020, and the one on the right is from the 12th of March 2020. Both of the clusters have 1 glinting satellite, so DirecTV 15 is on the left, and the one we're suspicious about, which is Spaceway 3, is on the right. These graphs show that it's not just one satellite that appears to be much more polarized, but the entire cluster. We compared the satellites in this cluster and found that the satellites have the same total intensity profile. You can see similar structures in the S0 for the non-glinting satellites, and if we were to zoom in on the S0 for the glinting satellite, we'd most likely find the same thing. This is also present in the S1s and S2s, but it is less apparent. So to make viewing the S1 and S2 a little bit easier, we separated them into 2 different graphs. This is for March 12th data, so still the corrupted data. It appears that the satellites in the cluster do in fact have the same structure in the S1 and S2. This should not be the case since all these satellites are different. And this shows that it's giving us information not about the satellites, but about the night of observation. Again, we noted that there were clouds that night, so this could be the source of the corruption. So looking at the time series, they show the data appears to exhibit the same dynamic variation. This points towards the theory that our data was corrupted, but we wanted to find a more quantifiable way of testing for this. So we decided to create scatter plots of S1 or S2 versus one satellite versus the S1 or S2 of another satellite in the same cluster. We then plotted the line of best fit, which got us the correlation coefficient, which was our R-squared value. This R-squared value is mainly what we use to determine if the data was corrupted or not. We did this for every satellite in every cluster to check the data, to check for corrupt data versus uncorrupt data. So here are some of the scatter plots from the corrupted spring 2020 data. This is not all of them. Since there were 4 satellites in the cluster, there are actually 12 graphs. Here are just 4 of them to show the data is clearly correlated. The R-squared values are between 0.49 and 0.71, which indicates that there is a correlation. The correlation would be even higher if I did a better job of clearing out some of the outliers. But this gives us a clear method of determining whether data has been corrupted or not. We're able to repeat this process for every cluster and found only one more night of data that was also corrupted. So now I'm going to go a little bit into the uncorrupted data. Analyzing the corrupted data is what I spent most of last semester on because it was really important to be able to distinguish between corrupted and uncorrupted data before we started analyzing the data thoroughly. My current project, what I'm working on right now, which I'm trying to make progress on, is starting to analyze the uncorrupted data. So most of the data that we collected was uncorrupted, and this is the data that we put through the same analysis as our corrupted data in order to test to see if it was corrupted. Here are some examples of uncorrupted satellite polarization signatures. As you can see, not all satellites have the same polarization signature, and not all observations are perfect. For example, the time series on the top left shows that DirecTV 15 glinted, but the Earth's shadow obscured the glint right before it was starting, and we could not observe the peak. Events such as this happen often during the equinox because of the solar phase angle. Sometimes a satellite can glint multiple times during an observation, which results in 2 peaks, which was observed in DirecTV 15, the graph in the middle left. You can see the double glint. In the uncorrupted data, the normalized S1 and S2 rarely drop below like -0.1 or rises above 0.1 unless it's during glint, like the graph in the top right. This is not observed in the corrupted data as it could vary between -0.4 and +0.4. Unlike the clusters before that I discussed, the cluster shown here exhibits low correlation. This is seen for all the uncorrupted data, not just this cluster. The R-squared values of the linear regressions are typically between 0 and 0.1, which points towards no correlation or low correlation. Some of the observations had an R-squared value a little bit higher, such as 0.15 or 0.22, but this was caused by small amounts of data points or not properly cleaning up images. We would expect slopes of 0 or infinity and an R-squared value of 0 between data that has no correlation. So future work, there's not too much more time that I have to do future work, but the biggest thing, so we've collected a lot of data. If we might get one more night of data after the equinox, not positive, but if we can, I'd like to probably observe one more night of data. And the big thing that I'm focusing on for the rest of the time I have is analyzing the uncorrupted data. So currently collecting a lot of satellite properties just on the internet, like bus type, launch dates, launch vehicles, just a lot of different satellite characteristics to see if we can make any conclusions about if there's any correlation between them. Some other things that probably won't be done by me, it could be done by me, probably not, is looking at the background degree of linear polarization. We maybe look at further analysis on data, which is variances, means, trends, And then the big thing, what we're doing is compiling satellite properties. Okay, are there any questions?
Great talk, Audra.
Thanks.
Who has some questions? Oh, you're getting rid of your slides already? Okay.
Oh, I'm sorry. I clicked something wrong.
Okay, Colonel Scoville.
Audra's very good presentation. Can you go back to the slide with all the different satellites? Yeah, that one. So I know that you were, mainly focusing on clearing out the corrupted data, coming up with a method to identify that, and you did a great job at that. Out of curiosity, with the limited time you've spent looking at the good data, and have you noticed any patterns or anything that might be useful in identifying the satellites or distinguishing characteristics or anything like that?
Yeah. So I don't think I've made any connections between certain polarization signatures and I don't know, bus type or anything like that yet. But the biggest thing that I've noticed by just looking at lots and lots of graphs is what happens during a glint. You can see a little bit probably in this one the best. You can see the S1 pretty much always will have this increase during a glint, and the S2 will always have this decrease during a glint. You can see it a little bit up here, but there's always this cross shape that happens during a glint. Most of the time, not all the time, sometimes it's a little bit lower and you can't really tell, but just looking at a lot of different graphs, I see it a lot. So I think that is an interesting thing that I've seen in the graphs, but I'm not exactly sure what it means yet.
Thanks.
Of course.
Thank you for your question, sir. Anyone else? So I guess, Audra, you listed a few satellite properties that you're looking at and exploring and doing research. Do you expect any would have a larger impact maybe in polarization signature, or if you had a hunch or a guess at what would play a role?
Well, based on not the research I've done, but people other than me have done in the past, they've been able to find a little bit of correlation between the polarization signatures and the size of the satellite, which I wouldn't be surprised if I could find the same thing. I would think material, but I don't know if we're going to do extensive research on different types of material, just because the type of material would change the polarization of the satellite, of the signature you see. But I'm not exactly positive if we'll even be able to find any sort of correlation.
So what about comparing glint versus non-glint signatures?
Yeah, so during a glint, pretty much anytime you get a big glint like that, it's going to be a reflection off the solar panels. So if we compare different types of solar panel material or size of solar panels or, I don't know, shape of solar panels, we could be able to make connections during the glint. And usually outside of the glint, maybe there's going to be more of the bus type and that sort of correlation. But I don't know.
So I guess when you're talking in and out of the glint, you mentioned previously that it was chosen that a glint would be defined as 15%, would be the start of the glint, I guess.
Mm-hmm.
Why was that chosen? Or I guess, could you explain a little bit more about that?
Yeah, so that was actually chosen by some of the cadets before me, and so that was just an arbitrary value I've been using. I've actually played around with it a little bit to see, I think I looked at orders of magnitude, so the minimum value, just raw count, to maximum, if it's a whole order of magnitude, I'd count that as a glint. And I tried that out for a little bit, and it actually seemed to work pretty well. I think there was a little bit of this one probably should have been a glint, but I didn't define it as a glint. And but just visualize looking at it, it was pretty accurate. So there are different definitions we've been playing around with using. 15% seems to be okay enough now, so I haven't had an issue with it.
Well, Audra, this is Dr. Chen. I can comment on where the source of that 15% came from.
Okay.
It actually came from Captain Josh Key when he was a cadet doing his spectral research, satellite spectral research with Anita Dunsmore. And he was the one doing the analysis of the black body comparisons between the black body curves and the satellite reflectance curves. And he decided, to the best of my recollection, he decided 15% was the demarcation for a glint versus non-glint time period.
Cool. So all my problems come from Captain Key.
that was Cadet Key.
Yeah, it was Cadet Key at the time, and yeah.
Now Captain Key.
Hey, Sandra, so how are you gonna take this research forward into the Space Force? Like, what are you doing in the Space Force?
Well, I'm just gonna be operating satellites and doing what I'm told. I don't know. I'd like to spend a little more time on this, but I don't know how much I'll be able to do while I'm, doing satellite operations.
Well, we're excited.
You can always do it when you're not on shift duty.
That's true. In my, large amounts of free time.
You won't be a cadet anymore, so yeah, you'll be surprised how much time you have.
Let's see.
Does anyone else have a question for Audra? Great. Well, thank you, Audra. Wonderful talk. And thank you for all the cadet speakers and mentors and whoever else was here to ask questions and challenge our cadets and inspire them, and have a wonderful weekend. Thank you.
Thanks.